Good morning, everyone, and welcome to the spring edition of the BioStock Life Science Summit 2023. We have a packed two-day program ahead of us with exciting company presentations and expert insights from financing experts and from Big Pharma. So I would like to say a big thank you to our event partner Nasdaq for their support, and also thank you very much for watching. Let's start the show.
Next up, we have Carbiotix, working in the field of microbiome healthcare. And here to present the company is CEO Christopher Cook. The floor is yours, Christopher.
Thank you so much. Christopher Cook is my name, CEO of Carbiotix. We're a company that's focusing on pioneering microbiome healthcare through the use of smart prebiotics. We currently have a rights issue that will be running through the 2nd of June this year, and there we're raising in total 12 million Swedish crowns. As well as a technical option in connection with this rights issue, where if fully subscribed, we'll raise an additional 15 million crowns. This rights issue is 100% secured through the management team, board, as well as external investors, and I myself will be investing in excess of 2.5 million crowns. For more details, please look at carbiotix.com/publications. So what is Carbiotix? Well, Carbiotix is an award-winning biotechnology company, as I said, pioneering microbiome healthcare, doing two things: prebiotic modulators and diagnostic testing services. Founded in 2014 as a spin-off from the Department of Biotechnology, we're operating currently 3 types of labs: a development lab for prebiotics, a microbiology lab, as well as a diagnostic service lab. In addition, we have a pilot production facility located in Bjuv, and we're currently 10 employees. So what is this microbiome? Well, it's the bacteria, viruses, and fungi that are living on us, as well as in our gastrointestinal system. And why are they so important? Well, as infants, we're born with both a high diversity and number of probiotic bacteria. And these bacteria play a significant role, among other things, in blood sugar and cholesterol regulation, mineral absorption, as well as linked to the risk of developing many different chronic diseases throughout life. And we know that around 70% of the immune system actually resides in the lining of this gut or gut tissue. We know as we go through life as well, both the number and the diversity of these bacteria go down, especially during states of disease. And we also know that prebiotics, or indigestible carbohydrates, are the food or main food source for these probiotic bacteria. But we also know that 90% of adults today are lacking between 50 and 80% of the soluble fiber or prebiotics they should be consuming in their diets. And as I mentioned, the benefits of prebiotics are quite numerous. But what's more exciting is the area of utilizing prebiotics to increase the pharmacokinetics, or how much active ingredient reaches the liver. And this is an area we're definitely exploring in Carbiotix. There is, however, a problem that the current generation of prebiotics have tolerance and efficacy issues. So what have we done? Well, we launched what we consider a superior product and companion diagnostic service. We've developed a prebiotic family called Carbiotix, derived from different cereals, such as oats and corn, as well as pea fiber as well. And this is up to 10 times more effective due to its complex structure. Now, this product can be used both in nutraceuticals and cosmetic applications, alone, blended, or in what we consider a co-intervention with medical foods and therapeutics. Now, to validate this particular product or the effect, we've launched a service called LinkGut. And today, it's the most effective, reliable, and flexible B2B microbiome test on the market. And when you combine Carbiotix with LinkGut, you have the smart prebiotic concept. Carbiotix will be sold exclusively to companies in the form of an ingredient. So we are a B2B company. And once approved, it will be priced at a premium given its complex structure. So essentially what we're getting paid for is the effect that we can deliver to the customers of our customers. Now, LinkGut today is sold as an effective B2B microbiome testing service, and that's done to wellness companies as well as health specialists, as well as suppliers of supplements and cosmetics. So how big is the market, the total addressable market? Well, the market we're targeting is in excess of 1 billion euros today, and we're targeting a 75% gross margin plus, actually. And the first market we're going to be targeting is the US market once we have GRAS approval, followed by the rest of the world, and then eventually Europe once Novo Foods is in place in 2025. Saying this, we think that the TAM, or total addressable market, can be upwards of 5x or 5 times with the introduction of symbiotic applications. That's combining the prebiotic with probiotics. In terms of the microbiome testing market, we're looking at a total addressable market of around 100 million today. But if we leverage the infrastructure and the testing service we provide today for clinical and research applications, that market is potentially 10 times more. In terms of key persons in the company, myself, CEO of the company since its founding in 2014. The suggestion or proposal right now is that I'll move over to chairman at the AGM on the 2nd of June. And Eric Diener, who I hired as head of sales and moved into the position of COO, will take over the position of CEO in the company. We also have 2 individuals who will be departing the board, and 2 individuals who've been recruited to replace Jonas Danielsson and Dr. Peter Falk. We have Geir Herstad as well as Jürgen Kampf. And both those individuals have been recruited to bolster or really bring up our ability to work with both sales and business development in the company. So in terms of the use of proceeds from the rights issue, as well as the technical option in 2023 and 24, as well as key milestones, within the Carbiotix area, we'll be upgrading our current site with both nanofiltration, ultrafiltration, and spray drying capacities. And there the purpose is to be able to produce these high-end products based on, as I mentioned earlier, corn, wheat, as well as pea fiber. And there we have 2 recently communicated partnerships, is the best way to explain it. One with a venture and the other one with Green Dairy. In this case, focusing on oat, and obviously there are other producers of non-dairy oat milk that are out there. What we also want to do with this scale up of our second generation process, which produces essentially twice as much yield as our first generation process, is to produce many, many different types of samples, both nutraceutical as well as cosmetic. And they'll be used both for formulation activities with customers as well as academic studies as well. So we want to start building up this research and study database demonstrating the efficacy of our Carbiotix product. On the regulatory side, we want to get GRAS self-affirmation for at least one nutraceutical product. And I say one there because there's a potential certainly to go beyond that, and ramp up sales as well through one or more partners. And again, that's part of our expansion strategy to build up our own production capacity as well as work with external partners. We want to do the same thing for our cosmetic products or ingredients. And there we want to start sales from Sweden. So starting to initiate sales, grow the production capacity in Sweden, and have those sales internationally from Sweden as well. Additionally, we want to make strides in terms of novel foods. So get feedback from this initial working group so we can meet our objective of getting novel foods in place by 2025. On the study side, we're soon going to be completing our initial metformin study used in conjunction with Carbiotix. And there we've been given already a green light to submit a more comprehensive or extensive application to get upwards of 2.5 million euros in funding to do an extended study. So our initial study was roughly 50 to 100 individuals. And now we want to get into a study where we have several hundred individuals and start including key partners in the supply chain. So pharmaceutical company, larger ingredient companies, and really build this out to a more comprehensive study. Additionally, the idea is that we'll be producing a much more efficacious Carbiotix product in this particular study. And the last point under at least Carbiotix related activities is we want to submit this first patent application for advanced symbiotic products. And we recently communicated a partnership with one of the top 10 probiotics companies in the world to develop symbiotic products. And we believe that this is a launching pad for this particular application, which could increase our total addressable market by as much as 5x, which I mentioned earlier. At the same time, we want to extend our link gut sales and partner network globally. We see this as a core cornerstone to our business as we integrate both the modulator as well as the diagnostic service to form this smart prebiotic concept. And last but not least, obviously, we have cost in connection with the rent of our production facilities, our development facilities, and our service provision facilities on the diagnostic side as well. And with that, I certainly welcome questions.
Thank you so much, Christopher, for that very interesting presentation. I'd like to start by talking about this collaboration agreement that you recently signed with this top 10 global player. First of all, what are symbiotics? I'd like to ask you that question first.
Well, you probably have to take a step back. We know that most people know what probiotics are because they're found in many different supplements and yogurts and other products on the market today. And probiotics are the bacteria themselves, and they're measured in counts. We know that what prebiotics are because we're working with prebiotics. So it's the food for the probiotic bacteria. Now, what you can do is combine both the probiotic, so the external bacteria, as well as the food which we produce, and together they form a symbiotic. Now, the benefit of the symbiotic is that you're introducing both the bacteria, which could establish itself either on an epidermal layer or in the GI system, as well as the food for that. So in theory, the idea is that you get a better establishment of this particular bacteria, which may be lacking in, for example, individuals suffering from a specific disease or indication area. Yep.
Well, in terms of the collaboration agreement itself, could you go into a little bit more detail about this agreement? I guess you can't really say who this global player is.
No, we're extremely happy with this agreement because it is a top 10 company, and my personal belief it'll be an impetus for other companies in the space to also become aware of the work we're doing, both in terms of our prebiotics, but also the diagnostic service, which, together form the smart prebiotic concept, it is a smart prebiotic concept. In this particular case, it's a company that obviously is in the consumer product space, and they have the ambition of developing these symbiotic products across different categories. And of course, we have to respect the anonymity request of this company, and we'll do that with any company that expresses an interest to do so. And that's for competitive reasons as well.
Sure.
So we're very excited about this. It allows us to validate our customization model for a probiotics company, which is great, as we continue to do that for different companies out there. And this is great. We're also going to be adding an individual in our microbiology team as well to support this as we build out that capacity in the company.
Well, Carbiotix seems to be in a transition phase, you could say, with some changes in management, board of directors that you mentioned. Yep. You'll be stepping down from your role as CEO and become chairman of the board. Yep. Well, first of all, how is that going to feel for you, stepping down from this role as CEO and becoming chairman?
I've been CEO since the founding of the company back in 2014. Since that time, I've obviously built up a lot of knowledge in this particular industry or branch, a lot of contacts, and that's my intention right now is to be a working chairman. Obviously, having a highly functional board is something that I think any company should have, and in our case, I wanted to certainly ensure that by bringing in key board members who would support a focus on sales and business development right now as we enter this phase. I've been working with this a long time now. So, 8 years of developing a new ingredient is not a short period of time. So I certainly wanted to make sure that as we transition into this much more intense commercial phase, that we have the right board to support that. And my expectations on the board are quite high as well. This is not, 4 meetings a year. This is us working together, for example, on a monthly basis, at least with key members, and then having a very intense follow-up and getting the support that can be provided through that facility. And then for myself, fundamentally, no big difference. I'll be leveraging that experience I have to focus on sales and business development. That's across the board. So with prebiotics companies, with probiotics companies, with cosmetics companies. And then my ambition as well is to consolidate a partnership with a leading company in the supply chain as well. That's my ambition for this period. And And obviously that will be there to strengthen our ability across the board when it comes to manufacturing and sales and R&D and legal, IP, etc. I think it's fundamentally necessary to find those types of collaborations out there.
And finally, as you go through this transition, what will be the top challenge for the new incoming CEO?
There's always an adaptation period, obviously. Nothing fundamentally is going to change. I will be there in person, supporting Eric in his role. He comes from a sales background, which I think any good CEO should come from. The ability to build relationships with different clients is fundamental for success in the business world. And obviously, there are a lot of practical issues in admin in connection with being CEO, and I'm there to support him during that transition as well. So my focus will be obviously working with business development sales and supporting Eric in landing in this new position as well. So I have no question whether he will, if he will be successful or not. I think that he will be successful simply because of the fact that he has the potential to be successful. He's demonstrated success previously, and certainly he'll have the support of myself as well as the board as well.
Mm-hmm. Well, thanks a lot, Christopher, for answering these questions and for your presentation. And we do hope to see you again soon.
Thank you so much. Pleasure to be here.
välkomnar vi Ulf Bladin från Prolight Diagnostics. Välkommen Ulf.
Tack. Visste du att årligen drabbas 23 000 patienter av hjärtinfarkt bara här i Sverige? Globalt uppgår det till närmare 3 miljoner som drabbas av en hjärtinfarkt. Och bara i USA så är det närmare 1 miljon som dör i sviterna av sin hjärtinfarkt. Mot bakgrund av det här så förstår alla och en var att det är oerhört viktigt att man tidigt kan diagnostisera och upptäcka när man misstänker en hjärtinfarkt. Det kan rädda liv. På Prolight Diagnostics här utvecklar vi innovativa patientnära testsystem som kallas för point of care-system för snabba, korrekta diagnoser av akuta tillstånd och i första hand för då hjärtinfarkt. Vår vision är att vi vill kunna erbjuda de främsta och mest innovativa point of care-systemen på marknaden. Point of care-system, vad är då det? Jo, det är alltså patientnära instrument där man snabbt kan analysera blodtester och ge säkra provresultat redan när patienten undersöks första gången. Och första gången det kan ju vara i en ambulans som ni ser här på bilden. Det kan vara på en vårdcentral, ett äldreboende eller på en akutmottagning på ett sjukhus. Bröstsmärta och misstänkt hjärtinfarkt hör väldigt intimt samman. Och det är som sagt kritiskt att man kan ställa en snabb och korrekt diagnos. Och under en hjärtinfarkt då utsöndras ett protein som heter troponin i blodbanan från den här skadade hjärtmuskeln. Det här troponinvärdet det hjälper doktorn att kunna bedöma om patienten har drabbats av en hjärtinfarkt eller inte. Och för att man ska kunna diagnostisera, eller som man säger på engelska, rule in eller utesluta rule out en hjärtinfarkt, då krävs det dels ett EKG och dels att man har ett troponinvärde. Och för att då med säkerhet kunna utesluta, alltså rule out en hjärtinfarkt, då krävs det att det här troponinvärdet kan mätas i oerhört låga koncentrationer. Och det kallas då för högkänsligt troponintest. Idag utförs de här högkänsliga troponinanalyserna på centrala sjukhuslaboratorier. Men där svarstiden då dessvärre kan uppgå till flera timmar. Därav så säger hjärtspecialister och vården generellt att det finns en väldigt hög efterfrågan av just användarvänliga point of care-system som snabbt och enkelt kan mäta det här högkänsliga troponinet med hög precision. Det finns således betydande vinster om man kan ha ett säkert point of care-system. På akuten då kan man då mycket snabbare utesluta roll out en hjärtinfarkt och därigenom kan man då frigöra sängplatser och också knappa resurser. Utanför sjukhuset då kan man ju få säkra testresultat redan när patienten undersöks första gången. Och därmed så behöver då inte ett blodprov skickas in till de här centrala sjukhuslaboratoriet som då medför en väldigt lång svarstid. Utan ambulanspersonalen och läkaren utanför sjukhuset kan snabbt diagnostisera eller utesluta hjärtinfarkten då tidigt i vårdkedjan. Och härigenom kan man också då prioritera de patienter som snabbt måste transporteras till sjukhus med ambulans. Så en tidig och korrekt diagnos kan rädda liv. Årligen här i Sverige så undersöks närmare 250 000 personer med bröstsmärtor och med misstänkt hjärtinfarkt på akuten. Men det är inte mer än 1 av 10, det vill säga ungefär 23 000, som får diagnosen hjärtinfarkt. Med det här så inser var och en att en tidig och korrekt diagnos det kan ju möjliggöra väldigt stora besparingar för sjukvården. För vi kan ju då undvika en onödig och lång sjukhusvistelse för de 90% som då inte har drabbats av en hjärtinfarkt. Och härigenom kan vi också göra stora kostnadsbesparingar och inte minst minska oro för patienter och deras anhöriga. Det är idag en växande global efterfrågan av dessa säkra point-of-care-tester. Och allt fler tester flyttas nu ut från de stora sjukhuslaboratorierna närmare patienten och den behandlande vårdgivaren. Och på den här bilden ser ni att point-of-care-tester omsatte cirka 34 miljarder dollar 2021 och förväntas öka årligen med 7,9% upp till mer än 70 miljarder 2030. Och för de här biomarkörerna inom hjärta, där troponin ingår, där ser ni att det omsatte 9 miljarder dollar 2021 och förväntas öka årligen med 9% till närmare 15 miljarder dollar 2027. Så med de här säkra patientnära testsystemen så kan vi alltså då ge effektivare sjukvård och också då spara pengar till lägre kostnader. 2022 då, det var ett väldigt transformativt år för oss på Prolight Diagnostics. Vi förvärvade och vi har också under året här nu effektivt integrerat ett brittiskt bolag, Cereno Diagnostics, som har då en helt ny banbrytande point-of-care-teknik för digital immunanalys. Jag kommer tillbaka till det. Vi har också tecknat ett kommersialiseringsavtal för vår andra plattform, den analoga PLD Microflex. Och det har vi gjort med vår utvecklingspartner, The Technology Partnership i Cambridge. Glädjande kan vi nu se att TTP nu under första kvartalet i år nådde en väldigt positiv milstolpe i ett utvecklingsprojekt inom distribuerad testning. Här kunde vi se att 2 laboratoriebaserade diagnostiska tester nu kunde överföras till plattformen. Och testresultaten från det här projektet indikerar att kommersiellt tillgängliga tester på de stora labben kan nu överföras till plattformen Microflex. TTP arbetar nu för fullt med att identifiera en lämplig kommersiell partner för att fortsätta den här utvecklingen som krävs för att ta tekniken till lansering. Men låt oss nu gå från dagens traditionella analoga teknik till en ny banbrytande digital point-of-care-teknik. Och vad är det då som är unikt med vår digitala point-of-care-teknik? Ja, istället för att jag pratar om det tänkte jag låta några av mina kollegor, nämligen Steve som är CTO, Eileen som är ansvarig för vår produktutveckling och vår dotterbolagschef Tobias berätta det för er.
The uniqueness of the Cyros technology is that it brings this amazing concept of single molecule counting into a very simple concept that can be developed into a product at very low cost and can benefit patients around the world.
The unique features of our Cyros point-of-care immunoassay are the ability to count individual molecules. This makes this one of the most sensitive immunoassays on the market. This will be done with a very small amount of blood. It will be simple, cost-effective and easy to use.
This technology is going to open up a completely new horizon in terms of diagnostics. Point of care until now has been very much hindered by the ability to go into very low levels of concentrations, and this is what this technology is going to open up.
Ja, som ni hörde från mina kollegor så kan vi alltså med vår digitala point-of-care-plattform räkna enskilda molekyler. Och det som är unikt är då att det här är bara en liten mobil analysenhet med ett väldigt lätthanterligt testkort som vi kan då tillverka till väldigt låga kostnader. En bråkdel av dagens kostnad jämfört med de stora dyra komplexa laboratorieinstrumenten. Dessutom kräver inte det här några dyra komponenter, dyra förbrukningsvaror. Stora fördelen är också att det har en väldigt kort svarstid och också att vi kan testa flera biomarkörer samtidigt som kallas för multiplex. Och allt detta kan vi göra med bara en enda droppe blod. Det här låter ju väldigt bra och nu kan ju vän av ordning ställa sig, men har ni gjort någon extern validering? Och svaret är ja, och inte av vem som helst utan från den kritiskt granskande myndigheten SBRI Healthcare i Storbritannien. Och det gjordes då inom ramen för Pioneering Medtech Innovations, där då vi tilldelades ett prestigefullt stipendium om ungefär 1 miljon pund. Och i den utvärdering som vi nu har gjort så har vi också kunnat visa upp för SBI Healthcare att vi har infriat och till och med överträffat alla de högt ställda målsättningar som lades i samband med detta stipendium. Också glädjande var att vi i slutet på förra året kunde visa upp Proof of Performance på vårt digitala point of care-system som var lika bra då som laboratoriekvalitet. Och det gjorde vi då med det sköldkörtelstimulerande hormonet TSH. De här låga koncentrationsnivåerna som vi kunde se är då indikativt för vad som krävs för att man med högkänslig troponin ska kunna utesluta en hjärtinfarkt enligt de gällande europeiska riktlinjerna. Och TSH är då ett väldigt välkategoriserat test och det har faktiskt lika storlek som troponin. Och fokus i den här studien var att vi ville optimera vår unika digitala point of care-teknik. Men resultaten var då en väldigt viktig milstolpe i vårt nästa steg i utvecklingen av vårt högkänsliga troponintest. Vi har också tittat väldigt mycket på marknadens högt ställda krav. Och jag är väldigt glad för att kunna meddela att vi infriar samtliga dem. I och med att vi har ett bärbart, enkelt system. Det krävs bara en droppe blod, kort svarstid, under 10 minuter. Som ändå kan visa väldigt hög känslighet och precision när vi nu detekterar biomarkörer i extremt låga koncentrationer. Och dessutom så kan vi då visa att vi fungerar även med multiplex. Och vi har som sagt väldigt låga produktionskostnader som i sin tur ger att vi kan ge ett väldigt konkurrenskraftigt pris. I och med att vi fick möjlighet att förvärva Syros så fick vi också de 4 grundarna till bolaget. 2 av dem hörde ni i intervjun och det som är väldigt imponerande är att de allihopa har disputerat. Men inte bara det, de har också jobbat hands-on med just utveckling av point of care-system och faktiskt tagit andra point of care-system hela vägen från ax till limpa och sålt ett system till Novartis för 1 miljard kronor. Det gör också att vi kan attrahera väldigt mycket kompetent personal till vårt bolag idag. Och utvecklingen sker därför i väldigt mycket snabb takt. Och det gör att vi under det här året har kunnat infria en rad milstolpar. Vi har som sagt kunnat visa proof of performance med det sköldkörtelstimulerande hormonet. Vi har uppnått alla milstolpar för den kritiska granskande myndigheten genom att vi lyckas också producera då 6 stycken fungerande prototyper. Vi har också valt ITL för att designa och utveckla nu ett toppmodernt kommersiellt instrument för oss för digital immunanalys enligt alla regulatoriska standarder. Och vi har också lämnat in ytterligare 2 prioriterade patentansökningar i tillägg till de 3 vi redan hade. Vi har nu framåt som avslutning. Nu ska vi ha ambition att sätta upp de första delarna av en utvecklingsproduktionslinje för vår testkortstillverkning. Och vi vill sticka ut hakan och hoppas kunna visa upp vårt koncept nu på den stora internationella kongressen American Association for Clinical Chemistry i USA här i juli 2023. Men sist men inte minst att vi ska kunna visa proof of performance nu på vårt digitala system med högkänsligt troponin. Jag ser här att jag har dragit över lite på tiden så därför så tackar jag så hemskt mycket för uppmärksamheten.
Och var hoppas du att ni befinner er om 2 år?
Om 2 år då hoppas jag att vårt banbrytande digitala point of care-system ska finnas på marknaden och att vi därigenom kan bidra till att man kan ställa tid och korrekt diagnos för alla dessa patienter som man misstänker har en hjärtinfarkt och därigenom rädda liv.
Tack för att du kom hit idag, Ulf.
Tack själv.
då har jag det stora nöjet att lämna över ordet till Lipum och vd Einar Pontén. Varsågod Einar.
Tack så mycket för det. Trevligt att få vara med här och jag ska berätta om vår utveckling av ett unikt läkemedel för behandling av kroniska inflammatoriska sjukdomar. Och som framgår av bilden så är det ju teamwork i allra högsta grad och vi har full fart framåt. Nu har vi kommit så långt så att vi är i klinisk fas. Vi har en fas 1-studie pågående och det är på vår läkemedelskandidat SOL 116 som är en humaniserad antikropp. Men om vi blickar lite bakåt så grundades Lipum 2010 och det baserades då på forskning vid Umeå universitet. Och för 2 år sedan så noterades vi på Nasdaq First North i Stockholm. Och om man nu ska sammanfatta Lipum så kan man ta en bild som visar vår målmolekyl. Och det är ett protein som en av Lipums grundare upptäckte för ungefär 50 år sedan. Det var i det sammanhanget att det var verksamt som lipas, alltså som enzym i bröstmjölk och hade betydelse för spädbarnets spjälkning av fetter och det näringsupptaget som barnet har från fetterna, eller från bröstmjölken. Och för ungefär 15 år sedan så upptäckte då grundarna av Lipum att det här proteinet förekom i förhöjda halter kopplat till där man hade inflammation. Och så att då blev man intresserad av det här och tittade på möjligheten att man kunde på något sätt påverka inflammationen genom att blockera det här proteinet BSSL. Så att man använde experimentella antikroppar och såg man att blockerar man proteinet så fick man mindre inflammation. Man kunde alltså tänka sig, hade hypotesen då att man skulle kunna använda antikroppar som läkemedel för att hämma inflammation. Och som ett resultat av det så startade då 2016 egentligen en utveckling av vår terapeutiska antikropp SOL 116. Och sammanfattar man vår patentportfölj då så kopplar den till det för att 2010 söktes det patent för själva principen att blockera det här proteinet och det är godkänt på ett antal större marknader och kommer ge oss skydd fram till 2030 och sen 2020 sökte patent för produkten, det vill säga antikroppar riktade mot det här proteinet och det ger oss skydd fram till 2040. Och vi ser att det finns många olika sjukdomar där det behövs, både för det medicinska behovet, en ny typ av verkningsmekanism. Vår huvudindikation är reumatoid artrit, alltså ledgångsreumatism, men vi ser också möjligheter för behandling av Crohns sjukdom, ulcerös kolit och SLE, där det finns stort medicinskt behov och väldigt stora marknader. Om vi går till reumatoid artrit till exempel så är marknaden idag ungefär 27 miljarder dollar och den domineras framförallt av så kallade anti-TNF-läkemedel, vilket också är biologiska läkemedel som hjälper många, men det är bara 40% av patienterna som har 50% förbättring på 6 månader och det är ju förstås inte tillfredsställande. Det behövs andra verkningsmekanismer som hjälper även de som inte har så bra hjälp för befintliga läkemedel. Så finns det en grupp sjukdomar som vi tittar på lite mer opportunistiskt, som vi inte lägger någon fokus på, men i den mån det dyker upp möjligheter så inleder vi gärna samarbeten och gör studier. Och vi har ju då vissa anekdotiska prekliniska data tidigare, till exempel på åderförkalkning. Ja, man hamnar inte i klinisk fas hur som helst och genom en slump, utan det är ju en väldigt lång och dyrbar utvecklingsprocess. Att för det första när det gäller biologiska läkemedel som en antikropp så krävs det ju först en design av antikroppen. Man ska utveckla celllinje och GMP-produktion och det tar ganska många år och kostar mycket pengar. Sen ska det göras säkerhetsstudier och det gör man ju både i provrör och på försöksdjur och parallellt med det så validerar man konceptet. Så att runda slängar 180 miljoner kronor för att komma fram till det läget att man kan starta en klinisk prövning. Och den prövningen vi har igång nu, det är en fas 1-studie i en så kallad SAD-del, det vill säga att varje försöksperson får en dos. Och den är ju helt och hållet fokuserad på säkerhet förstås. Säkerhet och tolererbarheten hos de här försökspersonerna. Och de första 5 grupperna som vi nu har tittat på som innehåller 8 försökspersoner i varje grupp, så totalt 40. så har vi inte sett några som helst negativa effekter eller biverkningar av SOL 116. Och då har ju dosen ökats för varje sån här grupp som har inkluderats. Parallellt med det så har vi tittat på farmakokinetiken och vi ser att vi har väldigt bra och lång halveringstid av SOL 116, vilket vi önskar ha, för vi tänker oss att i framtiden så ska det här vara någonting som man injicerar och tar en dos ungefär en gång i månaden, vilket är både praktiskt och behändigt för de som har sjukdomen reumatism. Men vi har i denna studie, lite ovanligt i den här fas 1-studien, så har vi också en grupp med 8 patienter som har reumatism, alltså RA. Och det vi tänker oss nu är att vi ska kunna studera hur halten av vår målmolekyl BSSL skiljer mellan friska frivilliga och patienterna. Vi vet att man har förhöjd halt av BSSL om man har reumatism och vissa andra former av artrit. Vi kommer också studera olika biomarkörer, alltså proinflammatoriska biomarkörer, cytokiner. Och allt det här kommer ge oss väldigt värdefull kunskap inför nästa steg och nästa studie, eller det som blir i fas 2. Men innan man slutför fas 1 så måste man också göra en så kallad multi-ascending dos, det vill säga då får samma försöksperson flera doser över tid. Och nu har vi sett att vi har så stabila resultat så här långt så att vi kommer kunna starta den här multi-ascending-delen betydligt tidigare. Så vi siktar på i alla fall nu att öppna upp protokollet, komma igång tidigare. Och då skulle vi spara både tid och vi sparar pengar så vi kommer fram snabbare. Och allt det här skulle inte vara möjligt om vi hade ett fantastiskt bra projektteam. Jag brukar skryta med dem, men nästan alla är oerhört erfarna och hängivna. Det är bara jag som inte kan de här proteinerna så bra. Nu har gänget förstärkts precis nyligen och det är styrelsen och det är Carl-Johan Spak med väldigt lång och stabil industrierfarenhet från läkemedelsbranschen. Och vi hälsar honom hjärtligt välkommen och vi glädjer oss över den förstärkning det innebär. Och teamet är viktigt, men det är också aktieägarna. Vi har väldigt stabila, intresserade och engagerade aktieägare, vilket jag är tacksam för, som med stort intresse följer vad vi håller på med och som stöttar den återkommande finansieringsbehovet som finns för att kunna driva det här vidare. Senast vi gjorde en emission var i oktober, det var en riktad emission i oktober i höstas. Det enda som inte aktieägarna kanske är så nöjda över är aktiekursen. Men tittar man på den lite närmare så ser man att sedan vi noterades i april 2021 så har vi i stort sett bara följt index för de hälsobolagen som finns på First North. Så att vi har inte överraskat varken negativt eller positivt, och det är väl så att makrofaktorer påverkar värderingen av hälsobolag väldigt mycket för tillfället. Däremot ser det inte likadant på affärssidan. Stora affärer pågår och det sker väldigt mycket affärer. Det här är en summering av affärer som är inom inflammation. Och man ser, om man tittar lite närmare, att det sker affärer redan i preklinisk fas. Bolag som köps eller licensieras och hela vägen upp till fas 3. Och ett som inte står med här är ju då förstås det MSDs köp av Prometheus för några veckor sedan. Då var det 2 projekt i fas 2 där de betalade $11 miljarder för det bolaget. Så att det sker stora affärer till betydligt annorlunda värdering. Vi är ju lite tråkiga i någon mening därför att vi har en strategi, vi följer den och vi kommunicerar den, så det kommer inga överraskningar. Vårt fokus ligger på den kliniska utvecklingen och i första hand då att genomföra de här säkerhetsstudierna som krävs i fas 1. Och så snabbt som möjligt komma in till fas 2 för att göra proof of concept-studier på reumatoid artrit. Men vi har inte lagt ner den prekliniska sidan utan vi fortsätter med prekliniskt utvecklingsarbete. Det gör vi för att titta på fler indikationer eftersom vi är övertygade om att SOL 116 kommer kunna användas för fler indikationer. Och vi gör studier för att lära oss mer om verkningsmekanismen och förståelse som också kan vara vägledande för att hitta nya indikationer. Och ett sätt att driva det här framåt är förstås att ha partnering av olika slag. Vi söker samarbeten för utvecklingsarbete och jag är övertygad om att det här på sikt så kommer det här ge tillfälle till eller möjligheter till väldigt intressanta affärer. Så att summerat igen så är det kliniska arbetet fokuserat på att slutföra fas 1 SAD-delen, och vi överväger att titta på möjligheten att starta den här MAD-delen där individerna, försökspersonerna får flera doser, komma igång med det tidigare. Men vi planerar ju också för fas 2, och vi har nu ett möte inplanerat med Läkemedelsverket för att ytterligare få bolla våra tankar kring utformningen av fas 2-studien. Och parallellt med det som sagt så fortgår det prekliniska studierna, och vi utvecklar också vetenskapliga samarbeten, och inom kort så tror jag att vi kommer vara klart med samarbeten som kommer vara fokuserade just på BSSLs roll i inflammatoriska processen och hur man kan blockera med vår antikropp. Själv är jag mest ute och träffar riskkapitalbolag och investerare, men vi har också märkt ett oerhört stort och ökat intresse för vår approach från läkemedelsbolag sen vi startade vår kliniska prövning i oktober. På något sätt så är det ju så för de stora bolagen att preklinisk fas kan bolag vara hur länge som helst. Man behöver bara en substans och en professor för att vara där. Men när man har kommit in i klinisk fas, då har man all dokumentation och man har gjort de investeringar som krävs för att kunna vara en partner som intresserar och bidrar till möjligheter för de här farmabolagen. Så att den typen av dialog pågår hela tiden, och de följer väldigt noga vad vi sysslar med. Så därmed ska jag avsluta och tacka så mycket för ert intresse och att ni har lyssnat. Jag har diverse länkar som man kan klicka på för att läsa mer, och man kan abonnera på vårt nyhetsbrev. Tack för intresset.
När du pratade av förklarliga anledningar mycket om studien här med er kandidat. Kan du berätta lite om varför ni har valt studiedesignen som ni har valt?
Nej, men det är klart att man har inte så stora valmöjligheter när det gäller en fas 1-studie. Och det är standard att den är randomiserad och att den är dubbelblind och så vidare. Men en sak som är ganska stor skillnad är att vi inkluderar patienter också redan i fas 1. Och det kan man läsa tillbaka i tiden, för så är det så att vi har tidigt så gjorde vi studier på patientprover och konstaterar att det är en korrelation mellan halten av BSSL i plasma och sjukdomsaktivitet. Och det gäller inte bara för reumatism, utan det gäller även för barnreumatism och psoriasis artrit. Så att där någonstans börjar ett intresse för att monitorera patientproverna. Och då planerade vi från början att vi skulle ha med patienter i den här multi-ascending dosdelen. Men det var ännu bättre om man kunde få in den i SAD-delen istället som man får tidigare. Och, varför är det bra då? Ja, det är ju för att vi kan jämföra friska frivilliga med patienter på olika biomarkörer och på vår målmolekyl BSSL. Och vi gör det i en och samma studie så att vi kan bilda oss en bättre uppfattning av hur väl sol blockerar BSSL och hur det påverkar olika typer av cytokiner, proinflammatoriska biomarkörer. Och det är ju information som vi kan ha nytta av i planeringen av kommande studier. Så att det var väl därför vi tog in det.
Om vi lämnar patienterna och det kliniska lite och istället pratar om partnering och möten med VC och så vidare som du pratade om här. Mm. Jag vet att du var på BioEurope, och jag vet också, eftersom jag snabbläste den, att i bokslutskommentaren så pratade du om gryende tecken på optimism inom biotekniksektorn. Ja. Vad tar du med dig från BioEurope? Var det där du såg dessa tecken?
Ja, men det är många saker. På BioEurope träffar vi ju riskkapitalbolag, alltså venture cap. Och det är ju flera av de här större life science-fonderna som har fyllt på kassan rejält i nya fonder nu, alltså mångmiljardbelopp. Och parallellt med det så skriver då det danska riskkapitalbolaget Sunstone Ventures, de är ganska givmilda med att dela med sina erfarenheter av olika slag, och de har gjort analys där de definitivt visar att värderingen nu som är i den här sektorn är nära det som man kan kalla historisk botten eller jämfört med tidigare data. Det är förstås inga garantier. Samtidigt har vi sett här också, vi ser affärer. Nu gjordes det ju mångmiljardaffär inom fältet inflammation precis nyligen. Men det gjordes också en förhållandevis stor affär här i Sverige på ett onoterat bolag i Helsingborg. Så att jag tycker alla sådana faktorer pekar på att det finns anledning till optimism, definitivt. Sen kan det ju bero på att det går så himla bra för oss och det bidrar till att man ser optimistiskt på tillvaron av det skälet.
Jag tänkte avsluta med att säga att du hoppas att Lipum kan dra nytta av den här optimismen då, men
Ja,
gör ni det?
Det gör vi redan, ja. Det gör vi absolut.
Tack så mycket för att du kom hit, Eden.
Welcome to the Expert Insights session of the BioStock Life Science Spring Summit, where we take a closer look at the big pharma perspective on partnering. Many small companies in the life science area are looking to strike a partnership. This could be through closing a licensing deal, selling a specific project, or even being acquired in an M&A transaction. All of this could be part of a general business model for the company. It could also be a preferred exit strategy for investors in the company. But how does a company become an attractive target for potential big pharma sharks looking for M&A opportunities? And once a deal is done, what happens after that? To answer these questions and a lot more, we've invited Anna Gran, who has loads of experience within M&A in big pharma and is currently at Novo Nordisk working as Associate Director, Corporate Integrations. Welcome, Anna.
Thank you.
It's so nice to have you here.
It's nice to be here.
Could you give us a short summary of your background and your current role at Novo Nordisk?
Sure. Currently, I'm in corporate integrations. And before that, I spent nearly I would say now nearly a decade in total in partnering. So meaning both collaborations as well as mergers and acquisitions and a little bit of alliance management in between for big pharma. And I've also spent a number of years in pharmaceutical product or drug launches, really looking at or working with what happens when you reach the other end of the funnel with a drug and it finally comes out and begins to hopefully make a patient impact. And before that, and this is really where my heart is, I was a research scientist. I worked in academia for nearly a decade in biomedical research.
How much of that experience as a scientist works into the partnership environment that you're in now?
Quite a bit. It's definitely a business environment where I have spent well over 10 years. But it helps when I'm working in a high science-focused, innovation, technology-focused environment. It helps to more quickly dive deeper into what is it that we're looking at? What is the possible impact that this innovation can have? What's the science behind it? And also really understanding it in a way that hopefully helps to ask the right questions, to evaluate opportunities, look at potential risks and de-risking plans. And then, of course, really also understanding how to best guide the teams to develop a plan to work together in the future.
Well, you've talked about your previous roles, and my curiosity is, how do you compare those previous pre-deal business development roles with your current role in post-merger partnerships?
They're siblings, I would say. Deal making is a sprint. Integration is a marathon, if you look at it from the acquiring partners side. And also, if you look at it from the side of usually then the smaller party, the biotech or the medtech company, deal making, it depends if it's a partnership. It can be a massive change for a small company, or it could just be an exciting project. Whereas if you're being acquired, while it might be one thing in the line of many for a big pharma company, of course, from a smaller company point of view, it is transformational. And in the space of partnering, I've of course had a broader role. So I looked both in search and evaluation. I worked as a scout for innovations. I have really led negotiations and due diligence for partnerships, and then also managed a couple of partnerships. But now in my current role, I'm really focused on the one kind of deal, which is the company acquisition, a full company acquisition, and how you then make both parts come together in the least disruptive way possible to create future value.
Mm-hmm. Well, you mentioned search and evaluation and your previous roles there. I'm curious to know, what did you look for specifically in a potential target when you were scouting?
Always compelling science first. And of course, I would not be the expert, but having a basic training in what is it that is that my company's experts are looking for. It helps to make the first assessment. And then it's really about if it's a scientific discovery, is the translation towards an innovation, so a real future patient impact, is that feasible? And then I would look at, well, where does this team want to take it that I'm talking to right now? What do they believe is the right way? What do they see as the best opportunity? And how do they see that they need help from a partner? Scalability. It's one thing to have a great idea and make a small number of products for one country. It's a whole different game if you're looking at the global market, which is, of course, what big companies would like to look at.
Yeah, sure. Well, and mergers often end with rather poor outcomes. What do you see in a company that would make you hopeful for a successful integration?
Well, yes, it's true. And I think there's a number floating around that around 70% of M&As do not capitalize or bring in the full expected value. And yet M&As happen all the time. So there's a reason for that. I would say, of course, the ground rule is if we like to think that we approach with expecting success from our own actions as well and our decisions and choices. So if we would not be hopeful for an integration, then we should not acquire the company. And those could be, of course, many reasons. A typical one would be the rationale is fuzzy. It's unclear if management teams cannot explain to their investors or their employees why it's the greatest idea in the world that these two companies come together. Then if that cannot be intuitive and easily understood by anyone, then that would be a warning sign. Sure. Of course, culture as well. People, M&A, it's not about just buying tangible assets like houses and labs and equipment and IP. It's also as much usually convincing humans that they should stay with us and continue to work with us. So that would be another thing that we'd look at.
And that brings me to my next question, actually, is how do you deal with these issues like company culture differences or a flight of talent, for example? How do you deal with these issues specifically?
Well, I like to say better every time and probably never as good as we desire to. I think that's probably true for every big company. As I said, it really comes down to explaining and creating an easily understood rationale for why this is a great idea. But then, of course, also retention risk you mentioned, talent flight. Why does that happen? It could happen. Usually it doesn't happen because of money. Yes, the money needs to be right, but we need to, as a partner, it's our responsibility then as the new owner to also, together with the team of the acquired company, make sure that we shape a new reason for why they should show up at work, what they should chase for, what's the common goal. And actually one of my key projects right now is in culture integration. So I'm looking at ways of designing a process and a toolbox for how we can more mindfully, as a big pharma partner, manage that journey of culture, meeting another culture and managing that change in a way that we're not stepping into cultural empirism as one big company, but also really making it work for both parties. That's very interesting, actually. Going into M&A strategy specifically, Big Pharma are busy as ever buying up assets to boost their own pipelines, not least Novo Nordisk, which made billion-dollar acquisitions in 2021 with Cereno and in 2022 with Pharmacolog. Would you say that Novo is putting more
efforts into partnering with external innovators?
Yes, for sure. We're known for being one of the leaders in the field of diabetes for a long, long time. In the recent years, we've also expanded into the broader cardiometabolic disease scope. And, of course, also expanded our footprint in rare diseases, and being able to access and really work with, the best of the best wherever that can be found in the world. That's what we want to do. And company acquisitions it's just one form, right? It's ultimately two partners coming together in a way that just makes the sum of the parts so much bigger. So for sure, that's what we are also, what you're seeing that we're already doing, I hope.
Well, you partly answered this next question, which is, do you still keep a keener eye on the Nordic markets, or do you find other markets more interesting?
Well, actually, if you look at the two recent acquisitions that you mentioned, and also other partnerships that have been announced in the past couple of years, they're all over the place. The acquisitions were in the US, so we are impartial to geography, I would say. We really go where the brightest minds are and where the best innovation can be found, and that includes the whole globe.
Speaking more broadly within Big Pharma, how do you approach the question of whether to build in-house capabilities versus acquiring those capabilities externally?
It comes down to a couple of factors. One is strategy. What is it that we're looking to do and move towards, and why? The second one is time. Do we have the time to build that in-house? And the third one is capabilities. Do we have the capabilities in-house to do so, to get where we want to be in the time that we need to be able to both capitalize on this amazing emerging science that's telling us to go in a certain direction, or, of course, to also stay competitive, which ultimately means bringing the best solutions to patients. So if we don't have, and many times we don't have the time, and companies don't have the time, don't have the expertise, and don't have the capabilities or the capacity, because they decide to go into a new direction and things need to move fast. And that's, of course, where the different forms of partnering step into the picture. And then it depends on what you're looking at, whether collaborations might be the best option. For example, to build a long-term pipeline, you might want to work with multiple collaborators. If you want to, as a company, make a big leap into a totally new area, including maybe a commercial portfolio, maybe a commercial team, so a sales force, then maybe an acquisition would be the best.
Looking at how investors look at things, they're usually quite good at sniffing out companies that are most likely to strike a partnership deal. Which features do you think usually give that competitive edge, let's say, to a company? I'm thinking about what you said about sniffing around and finding, we can only do so much. We do appreciate that companies step up and tell us and can convince us why they have the best idea in the world. So clarity of that message, clarity of why the science and innovation is compelling, why there are reasons to believe. That's the first step. And then we might have 10 minutes to hear you out, or we might have 2 hours, and we might have a million questions before we actually believe you. So I would ask for the patience there to just go through that. But once that kind of clarity of the science and the rationale and the story and the competitiveness is there, as well as this openness, a positive bias to have an open and honest conversation about collaboration options and really getting deep into what the opportunities are, that gets us both far along the way. So it's fair to say that communication is a very key part of partnering for sure in these environments.
It is, and I also worked in academia, and it is hard sometimes to step out of that space of world expertise and then explain to someone who is not an expert in the field why this is an opportunity that should not be missed. And it usually does help to have a sparring partner, a coach, external advice to get that the story in a succinct and clear way down.
Well, last question. What advice would you give to other companies that are looking to partner with Novo Nordisk?
Come find us. Come find us, and of course the main partnering conferences around the world. But also come find us on our website. We do answer. I can almost promise we do answer to requests as well, through email, through the partnering website and the portal that we have.
Well, thank you so much, Anna, for answering these questions. These very interesting conversation about partnerships within life science and with big pharma, and we really appreciate your time today.
Thank you so much. It was a pleasure to be here.
Next, we are showcasing Cereno Scientific, who are developing novel treatments for cardiovascular disease. Here to present the company is the CEO, Sten R. Sørensen. Welcome, Sten. The floor is yours.
Thank you, Mike. Appreciate it. So, pleased to be here and to update you on our journey with Cereno Scientific. We have a vision to develop innovative treatments to prolong life and improve quality of life for patients with common and rare cardiovascular diseases. Now, we have our main platform on epigenetic modulation. I'll get to that. We have 2 programs, one in phase 2 in the orphan disease, pulmonary arterial hypertension, and it's aimed to be completed by the end of this year. We have a novel HDAC inhibitor, CSO14, at the end of preclinical evaluation and documentation, aiming to go into phase 1 for thrombosis prevention beginning of the next year. Our third program is a IP receptor agonist, CS585, recently in-licensed for global development and commercialization from University of Michigan. Our lead program in phase 2 is pursued in 9 clinics. Our study on PH patients, 30 patients to be completed by the end of the year. So only in the US. We are based in Gothenburg, but we have a daughter company in Boston, US. We also have our preclinical programs pursued in collaboration with University of Michigan in the US. So we have a lot of research going on in the US. Our epigenetic platform, epigenetic modulation, is the 2 HDAC inhibitor programs, CS1 and CSO14. And by that program and those molecules, we modulate the expression of important proteins for man that sometimes can go the wrong way in their production or concentration in the body, and then you develop diseases. So we have these 2 programs in 2 different diseases targeted for better therapy for these patients. We have a very strong scientific advisory board, mainly based in the US, that have supported us in our journey and have added strategic insights and scientific insights to our programs. In addition, they have produced white papers, so to speak, on all the important research behind our HDAC inhibitor platform. 100 articles, 100 references were published in The Lancet, one of the most prominent scientific journals in science. We have done an evaluation early on here what we should focus the HDAC inhibitor platform on, and we defined pulmonary arterial hypertension, the orphan disease, to be the first target. And the second target for the second program, CSO14, is aimed at thrombosis prevention without increased risk of bleeding, something very much sought after. The reason the HDAC inhibitor program is so interesting is that it looks like it can have not only prevention of thrombosis without increased risk of bleeding, but also seems to impact the disease progression, slowing it down or even reversing it in cardiovascular disease. We have several studies in pulmonary arterial hypertension and in other diseases showing those effects. So there are multiple indications we could pursue, but we pursue PH and thrombosis prevention with our first 2 programs initially. So where are we? CS1, as I mentioned, is at the aiming to conclude phase 2 program by the end of the year. We will get top-line data on the effects of CS1. The study is mainly safety and tolerance, but we're looking for efficacy parameters in 3 different dosages. CSO14 is at the end of preclinical evaluation, and we are aiming to submit an IND by the end of this year and go into man early next year. CS585 is being evaluated. What indication is best for that compound? So let's look quickly at CS1, our HDAC inhibitor for pulmonary arterial hypertension. This disease affects the vessels in the artery system. They get too narrow. There is too much pressure to pump against for the right heart, and eventually the right heart grows and fails. So these patients die prematurely and have a very bad quality of life prognosis. The current drugs are old-style drugs. They vasodilate, they lower the pressure in the artery system, pulmonary system, but they don't do anything really on disease progression, and that's where we come in. Everybody is looking for this in the field of science and this disease, and based on these parameters to the right here that we have documented in preclinical work, pulmonary pressure reduction, reverse remodeling, antifibrotic, anti-inflammatory, and in addition antithrombotic, this agent, HDAC inhibition and CS1, fits like a glove for this disease, and we are very excited to see the data eventually. Another company that pursued disease modification with a drug called sotatercept was bought by Merck 1.5 years ago for $11.5 billion and that actually signifies the interest in the sector and these kinds of effects from new drugs. If we look at our drug in preclinical documentation and look at what sotatercept, the drug that was bought by Merck, had for what kind of documentation was there in preclinical, it's very, very similar. It's two different drugs, two different mode of actions, but the data is similar. So we are very excited to see what we will get out of our clinical program. How does the clinical program look? Well, I mentioned the preclinical work and the publications. We've been in phase 1, now we're in phase 2, aiming to complete by the end of this year. We're doing that trial together with Abbott, major healthcare company. And the reason we're doing it is that they have a cutting-edge technology to measure pulmonary pressure, an implantable device that the patients actually get to keep for life. But that device can measure how many times you want and can also measure cardiac function. So it's very good to have that in this study. And this is really the future of diagnosis and therapeutic intervention. So our phase 2 study in pulmonary arterial hypertension with our drug CS1 is aiming to include 30 patients. And the latest progress is that we had 16 patients enrolled, 9 patients received CardioMEMS, 5 patients randomized on drug, and 2 patients actually completed the study. And the design you see here, the patients are randomized after a CardioMEMS implantation out to 3 different drugs, and they're treated for 12 weeks. And we are able to measure a number of times with CardioMEMS. And at the end, we'll evaluate a number of parameters in the study. We are measuring a number of effects here. The primary endpoint is safety and tolerance, but we are measuring cardiac function through CardioMEMS and pulmonary pressure. We are doing echocardiography, we're doing MRI, and we're doing all the classical measurements that are done for approval of this kind of drugs, such as 6-minute walk test, quality of life, etc. So we are hoping to get a lot of interesting data out of this study. Let's move on to our next HDAC inhibitor program, aiming to get into man next year in the spring for thrombosis prevention. Thrombosis, cardiovascular disease is major. It's the biggest problem in health for man, and it's also the most common cause of death. 80% is blood clots, so thrombosis formation, blocking the blood flow in the arteries or veins and causing damage to major organs such as the brain and the heart. Now, all drugs that are used there are weakening the coagulation system. Antiplatelets, anticoagulants, they do that. So they have an effect, but the downside here is that there's a quite major risk for severe bleedings. So out of 100 patients treated with these agents, 3 actually die. And out of 100, 25 get some kind of bleeding. So there is a tremendous need for effective therapy that doesn't cause bleeding risk. And that's where we come in. Just look at this picture here. This is some of the bleedings that are not fatal. Many of them require hospital care. Okay, so in comparing the other agents here, you see CSO14 to the right here, that we have the ability to prevent thrombosis documented in animals. But in addition also, there's documentation on organ protection, as I mentioned earlier, for HDAC inhibition. We are evaluating this compound and documented in collaboration with University of Michigan. And here's an example of the animal work that has been published at the world's largest cardiovascular congress, European Society of Cardiology in Amsterdam. And you see it on the photos below. That's our agent preventing clot formation. Our last program, CS585, is an IP receptor agonist, and we just licensed that from University of Michigan. It has the ability or potential to be best in class of these agents, and we are documenting where to apply this, what disease, and it's 1 year behind CS014. So that actually concludes my presentation. 3 programs in cardiovascular disease, 1 in rare, 1 in common, in HDAC inhibition, and then the novel IP receptor agonist. We are seen all around the world in presentations and publications, and that's strengthening our brand name and also our exposure to potential partners and big-time financing groups.
Stan, thanks so much for your presentation. What makes your pipeline innovative?
We are pursuing HDAC inhibition, epigenetic modulation through that mode of action for cardiovascular disease. And that's novel. It hasn't been applied forcefully in the clinic. And it's new. Epigenetic modulation is a novel approach in medicine. So we have 2 programs there for 2 different diseases.
Mm-hmm. You speak in your presentation of your drug candidates having disease-modifying potential, which obviously is related a little bit to this epigenetic modulation that you talk about in part. Could you describe in more detail what that means, disease-modifying potential?
Well, in cardiovascular disease, you see a progression of changes in the vascular structure and also in muscles, etc. So the vessels that transport our blood, which is critical for oxygen supply, but also for various other things, gets more narrow and get fibrotic, and it impacts the ability for heart to pump, and then the heart gets hypertrophic. So we now know there are inflammation processes involved in this, and there are a number of various markers that drive this change. And we have seen in animal work that we can slow down this kind of progression of, for instance, fibrotic development or hypertrophy in vessels and heart. But we've also seen and documented that we can reverse this beyond the actual dynamic reduction of systemic or pulmonary pressure, for instance. And so this is very important for cardiovascular drug therapy and for the patients in the future. And that's what you want to get at. Yeah.
And finally, Cereno is shifting from Spotlight to NASDAQ First North. What are the implications here? Why are you making this move?
Yeah, it's for us it's obvious. So it's been said that it's a maturing issue for biotechs. We feel that we have now 3 programs. We're nearing completion of a phase 2, very important study for us in PH with CS1, and we want to attract international partners and investors and the industry, and NASDAQ has a very good brand name, and it's also allowing foreign investors to a higher degree to invest in our company. And it's been a request from venture capital groups or financing groups that we have met over the last year and exposed the company to. So it's a natural move for us.
Mm-hmm. Thanks so much, Stan, for answering the questions and for your presentation, and we hope to see you at the next event.
It's been a pleasure. Thank you.
now we will listen to a presentation from Erik Digman Wiklund, CEO at Circio. Welcome. Go ahead.
Thank you very much. Great to be here at BioStock, and I'm glad to present Circio. Our forward-looking statement. RNA is emerging as a very interesting therapeutic class. In the last 2 years, we saw the approvals of the first mRNAs with the COVID vaccines. And these drugs have rapidly emerged to be the top-selling drugs overall in the pharmaceutical market. The 2 mRNA vaccines from Moderna and BioNTech sold for more than $50 billion last year alone, which is remarkable. It puts them in the top 5 of all drugs combined. So this proves, finally, that mRNA or RNA in general is an efficient therapeutic. It's clinically validated, it's logistically validated, and it's now been shown to commercially also be viable. This is a massive breakthrough for RNA therapeutics. However, mRNA still faces several challenges that have held back development, and it's also holding back the potential for the future. The biggest drawback of mRNA or linear RNA in general is that they're highly unstable. They get degraded fast. They have a short half-life. And this short half-life, it complicates delivery. It's hard to get the RNA to where it needs to go, and it's hard to keep it there for long enough to do its job. We believe that circular RNA can overcome several of these challenges and really can be the next frontier in RNA therapeutics. Now, let me tell you why circular RNA is so exciting. So circular RNA, by being circular, it lacks a free end. Now, mRNA gets broken down by what we call exonucleases. In this slide, you can see them as these small Pac-Mans. They eat up the RNA from the ends. A circular RNA doesn't have an end, so the Pac-Man doesn't have anything to latch onto. This means that the circular RNA is resistant to the RNA degradation mechanisms in the cell, and it massively improves the half-life. Due to this advantage, you can now have a much more stable RNA species, and you can achieve also several other functionalities that simply are not doable with mRNA. So we view circular RNA as a toolbox to create a completely new class of medicines. And that's precisely what we're doing at Circio. Our science is led by Dr. Thomas Hansen. Dr. Thomas Hansen, together with myself, we are actually the discoverers of circular RNA. Back when we did our PhDs 15 years ago, we stumbled across circular RNA looking for something else, and we published the first paper on circular RNA. Thomas then built a research career in an academic group studying circular RNA and really has emerged as probably the most experienced circular RNA scientist in the world. And we have now fortunately been able to recruit him to Circio, and he is setting up our circular RNA program. We're not the only ones that are interested in circular RNA, believe this is a future very exciting therapeutic class. This graph here shows the financing deals that have been done in the past 5 years for RNA therapeutics. You know, the biotech market was in quite dire situation the last couple of years. There has been very few financings done, but actually the RNA therapeutics financing market has been strong. In 2022, which was a very difficult year for biotech in general, RNA therapeutic financing was higher than pre-pandemic levels, more than double compared to 2020. So RNA therapeutics is actually one pocket that is still doing well in biotech. If you break down the RNA financings, it turns out about 40% of all the RNA deals that were done were for circular RNA concepts. So this is not something we are just making up. This is really where the momentum is at the moment. There has been several deals and large companies launching. We generated this slide here to show the momentum. 2 famous companies in the space called Lund and Orna Therapeutics launched with massive Series A financing. Orna did a deal with Merck. This all happened in the span of the last 12 to 18 months. And then just a few weeks ago, another company, Orbital Therapeutics, launched in the US with a Series A mega round. So this space is really moving fast, and we are planning to latch onto this and be a player. And we're doing things differently. These companies are doing synthetic circular RNA. It means they're effectively doing what BioNTech and Moderna did, LNP packaged circular RNA that they utilize as a messenger. So it's a repeat of the mRNA playbook, just using circular RNA. We're doing it differently because we are using a vector delivery approach. We are generating DNA-based vectors that contain the recipe to generate circular RNA intracellularly, in situ in the patient. So the product we're making is DNA. We give this DNA to the patient, and it contains the recipe for the patient to make its own circular RNA. No one else we are aware of is deploying this technology for circular RNA therapeutics. To explain this a bit more granularly, the core of our technology is what we call circVec. In this little schematic, you can see it as this genetic cassette. It's a piece of DNA, and this piece of DNA carries the instructions, the recipe for the cell for how to generate the circular RNA. And then the circular RNA is formed, and we've engineered the circular RNA in a way that it's very stable. It's very efficient at protein translation. It already outperforms mRNA, and by virtue of its stability, we've built in several additional regulatory functionalities as well. We have filed IP for this core construct, and we have this nicely covered, and it's agnostic to what vector you're using. So the circVec cassette can be deployed in any DNA-based vector system of your choice. We are focusing primarily, or initially by using adenovirus as a delivery platform, and this is the second step of our IP platform, is the utilization of this circVec cassette for circular RNA generation from adenoviral vectors. We have already demonstrated that our circular RNAs are much more stable than mRNA and that they produce more protein than mRNA. You can see here in vitro experiments, we have achieved a 15 times extended half-life of circular RNA versus mRNA. On the right-hand side of this slide, we show you some data on the protein expression. And what we're seeing is that even with lower copies of circular RNA present, we can make more protein. A circular RNA beats an mRNA on a head-to-head basis. And then as expected, if you compare 48 hours to 96 hours, over time, the circular RNA accumulates, whereas the mRNA degrades. And we anticipate this to grow, the difference to grow further over time, so that the RNA, the circular RNA, will be persistent and keep producing the protein in a much more stable manner than you can achieve with an mRNA. If this works in patients, it will be a massive breakthrough, opening up completely new territories for what you can do with RNA therapeutics. We are currently replicating these data in vivo, and we expect to be able to share in vivo data by Q3 this year. Here we compare our technology to some of our competitors. Lund and Orna I mentioned before, they're doing the synthetic circular RNA approach, and Moderna and BioNTech, they're doing synthetic mRNA. I'd like to highlight a couple of advantages. One major advantage is that by using our adenoviral system, we can reach solid tumors. We can get circular RNAs delivered into cancer cells. And this is simply not doable with the approach that these other companies are deploying. So we believe we have a unique angle in oncology to get circular RNA into solid tumors, and this will be the first program in the clinic for a circular RNA therapeutic in cancer. In addition, we are building on vector technology, which is already existing. The manufacturing is already existing. We can quickly plug and play this into our existing setup. Circular RNA manufacturing simply does not yet exist. So the manufacturing needs to be set up and it needs to be scaled up. And this will take significant investment and significant time. So again, we think we have a head start here by deploying vectors that are more easy to manufacture. We can be quicker to the clinic, and we can get to the clinic in indications which are currently not possible to approach using conventional RNA technology. Let me give you an idea of how we're thinking to deploy this. We see this as a platform with broad potential. The first step, as I've said before, is oncology. We're building on existing legacy technology in the company. We have an adenoviral delivery system that it's already been in a large number of patients. We're re-engineering this for circular RNA delivery into solid tumors to enable long-term and durable expression of proteins inside of cancer cells. This we aim to have in the clinic already in 2025. The second step of our development plan is in vaccines. In vaccines, we are planning to use the power of adenovirus together with a circular RNA to make potent single-dose vaccines that really can be a transformational vaccine concept. This we're planning to develop preclinically and then out-license to a collaboration partner. And eventually, we want to move this into rare disease. We think circular RNA has a particularly compelling benefit in rare disease setting where you need to replace proteins, because these transcripts are highly stable. They can give durable protein expression and deliver and replace a malfunctioning protein over time. Again, something which is not doable with a very unstable mRNAs. So we think over time, maybe the biggest potential lay in rare disease, and we're planning to expand into this area in the short-term future. The concept looks like this in cancer. So we're generating adenoviral vectors that we inject directly into solid tumors. They carry then the recipe for making circular RNA to the cancer cells. The circular RNA is produced in the tumor, and the circular RNA then express the protein of choice we want to deliver, and it exerts additional regulatory functionality to have multifunctional, highly targeted anti-cancer therapeutics. And again, the first candidate here, we expect to bring into the clinic already in 2025. Similarly with vaccines, we have a DNA-based vector, carries the circVec cassette. We express then the antigens from the circular RNA, which gives you prolonged durable expression of the antigen, and thereby a stronger and more durable immune response. In rare disease, we've done an extensive screen, filtered existing disease or monogenic disorders that we believe can be targeted with our approach. And we have arrived at a short list of 5 disease areas that we are prioritizing for further evaluation. And we anticipate to start in vivo work in the second half of the year and present data within 12 months of a proof of concept in a rare disease setting. So overall, we think we have a unique edge in this emerging field of circular RNA. We have world-leading experts on board. Our team is led by probably the most experienced circular RNA scientist in the world, Dr. Thomas Hansen. Our delivery platform opens new avenues for how to use circular RNA. We can use it in oncology, we can use it in vaccines. This is already ongoing. We're starting in vivo work, and then eventually we're moving into rare disease, which may be a massive opportunity for the future. And we aim to be the first into the clinic with a circular RNA product that will be in oncology, and we hope to achieve that in 2025. So with that, I wrap up the presentation, and we're ready to take some questions.
Thank you so much for the presentation. And many of our viewers might know you by your previous name, Targovax. So I was wondering if you could just make a quick comment about this recent change.
Yes, correctly. We recently changed our name from Targovax to Circio. The new name reflects our efforts in circular RNA. This is now the #1 strategic priority for the company. And Circ is for circular RNA, naturally, and IO is for immunotherapy, immuno-oncology. This reflects our heritage as an immunotherapy company. That's where we come from. And we also intend, as I've described, to move into oncology in an immunotherapy context going forward. So we designed this name to point both forward and also stay consistent with our legacy.
And as I understand it, you're seeking for a partnership. What kind of companies are you looking for?
We are looking for partnerships in multiple avenues. We can use our platform to deliver therapeutic proteins of choice. So we're looking for partners with interesting therapeutic proteins that could be efficiently delivered from our vector system. And this can be agnostic of therapeutic area. It can be in oncology or other contexts, but sort of making on-demand constructs for partners. That is one avenue. The second avenue we're intending to partner is in vaccines. Developing vaccines clinically is a large undertaking. We believe it's probably better to out-license our vaccine concept to a vaccine company. So our intention there is to develop a preclinical proof of concept and then seek partnerships to bring that into the clinic. And I think that should be achievable in maybe already in 2024.
We could end this by looking forward. Where do you see the company in 5 years from now?
In 5 years from now, I see Circio as the dominant player in vector-delivered circular RNA. We want to be a key player in the circular RNA space overall, and we want to have a versatile platform that is validated in multiple settings with our vector delivery approach of circRNA.
I see. It will be interesting to follow your journey. Thank you so much for coming.
Thank you. Thanks for having me.
Nästa presentation kommer från Anders Hägerstrand, VD på Annexin Pharmaceuticals. Varsågod.
Tack så mycket. Tack för inbjudan att presentera bolaget för alla lyssnare och tittare. Kortfattat så är bolaget då publikt på First North. Vi är Stockholmsbaserade. Vi är semivirtuella kallar vi oss. Vi är bara 4-5 personer på ett kontor här i Stockholm, så vi köper tjänster runt om i Sverige och i världen. Vi är experter på Annexin A5 som är ett humant protein som finns i alla celler. Vår version av Annexin A5 heter ANXV. Det är i princip samma sak när jag säger Annexin A5 och ANXV, fast produkten som vi utvecklar heter ANXV. Vi producerar den enligt alla konstens regler. Vi har patent på produktionssystemet och produkten som kommer i det systemet som är väldigt tajt, väldigt brett. Vi har patent också på att använda det här ANXV och andra Annexin A5-versioner, humana, för behandling av sjukdomar. Av alla möjligheter som finns för Annexin A5 och ANXV så har vi valt retinal venokklusion, en ögonsjukdom, med en marknad på över $20 miljarder om några år. Väldigt stor, kommersiellt, väldigt ganska okänd medicinskt. Vi har också nu senaste tiden valt att fokusera även på cancer och tagit en del steg in i det fältet där vi kan bli också en first in class typ av läkemedel inom immunonkologi och läkemedelstargeting. Det team jag har med mig, först ska jag säga om mig själv, jag är läkarforskare från Karolinska, jag har varit på AstraZeneca, så en del småbolag som vd och forskningschef. Jag har varit med och tagit läkemedel från fas 0, i djurstadie, in till patienter i fas 2. Och varit med och sålt några bolag och tagit licenser och givit licenser. Susanne Andersson är nytillträdd CFO från publika miljöer också. Och kom in i bolaget helt nyligen med väldigt god erfarenhet. Susanne Sachdev är clinical operations och operativ chef hos oss. Erfarenhet av kliniska studier och i stora bolag och mindre CROer. På den medicinska sidan så Anna Frostergård som är grundare och kan allt om Annexin A5, har varit med i bolaget sedan vi kom in på börsen 2017 och även dessförinnan. Jag är oftalmologisk, det vill säga ögonläkarspecialist i Maruf Sadni, som varit med och tagit fram läkemedel på Novartis och på Allergan. Vi har också Alain Thibault som är immunonkolog, utbildad i Kanada och USA, som är vår stöd i vår cancersatsning. Biologin då? Ja, Annexin A5 och ANXV binder till PS, fosfatidylserin. PS sitter på insidan av celler, där celler är sjuka, blir skadade på annat sätt annorlunda, så skickar man ut det här PS på utsidan av cellen. PS är sin tur signalerar till omgivande celler på alla möjliga olika sätt. Det kan orsaka klibbighet som i RVO, det kan orsaka en immunreaktion eller frånvaro av immunreaktion som i cancer och i andra sjukdomar och även andra saker. Annexin A5 är kroppens sätt att modulera och balansera vad PS gör i kroppen kontinuerligt. Vi har fokuserat på retinal venokklusion. Den sjukdomen är en propp i bakdelen av ögat, näthinnan. På näthinnan ska ljusimpulserna omvandlas till en nervsignal som sedan blir en bild bak i hjärnan. Därför är näthinnan väldigt känsligt organ. Får man en propp där, vilket är grunden till den här sjukdomen, så får man backflöde, det svullnar, näthinnans lager separerar från varandra som ni ser på den svartvita bilden där i mitten. Man får blödningar, celler dör, synfältet blir avsevärt försämrat, man ser blurrigt, man kan bli helt blind i vissa fall. Och den här sjukdomen är svårbehandlad. Vad har då PS med det här att göra? Man har visat att röda blodkroppar hos de här patienterna uttrycker PS på utsidan, vilket de inte ska göra. Vilket leder till en klibbighet. Och att de fastnar på varandra, de fastnar i små blodkärl. Och Annexin A5 och därmed ANXV kan blocka den här klibbigheten. Och det här visades av franska forskare. Och vi tog licens till den användningen av Annexin A5 för behandling av RVO. Dagens behandlingar ser ut som ni ser på till vänster. Det vill säga man skickar in med nål anti-VEGF-terapier rakt in i ögat. Det sker inte bara en gång, det sker 2, 3, 4, 5, 6, ibland flera år. Månatligare, kanske var 6-8 vecka, får man de här injektionerna. Och det är förstås obehagligt och kostar mycket pengar. Vi erbjuder ett alternativ, hoppas vi, som innebär att man får en intravenös infusion i handen eller i armen ett antal dagar efter att man får sjukdomen. Och sen ska man i bästa fall få mycket mindre behov av sådana här anti-VEGF eller inte behöva dem alls. Det är målet. Och vi är lite annorlunda än anti-VEGF. Anti-VEGF dominerar marknaden totalt, kommersiellt och på alla möjliga sätt. Det är egentligen bara en behandling av komplikationerna till RVO. För får man RVO får man svullnad och man får kärlnybildningar som då anti-VEGF är bra på att behandla, förhållandevis bra. Men ANXV erbjuder en massa andra mekanistiska fördelar jämfört med anti-VEGF. Och vi möter faktiskt väldigt stort intresse för vårt projekt. Vi fick ett Innovation Award på en konferens borta i San Diego i vintras. Vilket vi förstås är väldigt glada över och är glada över i konkurrens med 40-tal andra bolag. Jag har nyligen presenterat data där vi har fluorescensmärkt, satt en liten fluorescerande ämne på vårt ANXV injicerat i patienter med nyligen diagnostiserad RVO. Så har vi följt den här fluorescensen in i ögat. Man kan titta in i ögat med sofistikerade kameror som är väldigt avancerade och bra. Och då ser man blodkärlen som ni ser uppe till mitten lite grann, sin vackert kärlträd. Och sen kanske svårt att se, men på A och B där ser man små gula pilar. Då är det vårt fluorescerande ANXV som har nått fram till den ven som ligger i området, den här runda cirkeln som är påverkat av den här proppen. De läkare som genomförde den här studien kunde även visa att patienten hade PS på ytan av sina röda blodkroppar. Så det stämmer med hypotesen och det stämmer med vad vi vill kunna åstadkomma. Det mest spännande kanske är att vi har en pågående fas 2-studie i USA. Fas 2A, Proof of Concept kallar vi den för. Det är ett ganska fåtal patienter, upp till 25. De har inte fått anti-VEGF. De behandlas med placebo eller med Annexin i 5 dagar. De följs upp intensivt på många olika sätt. Med vanliga såna här syntavlor som ni kanske har testat allihopa. Men även med massa olika undersökningar. Vi tittar på blodflöden, blodkärlsstrukturen, hur näthinnan fungerar på andra avseenden. Om de behöver anti-VEGF trots behandlingen, Annexin versus placebo. Och om de patienterna har PS på ytan av sina röda blodkroppar. En intensiv studie. Vi följde dem i 29 dagar. Därefter går de ur studien. Och sen ska vi förhoppningsvis avsluta första kohorten med 8 patienter nu inom någon månad. Immunonkologi och onkologi. Så att onkologi är ett stort ämne. Vi vet att cancer behandlas med strålning, med cellgifter, med kirurgi. Och det är fortfarande den näst vanligaste orsaken till att människor dör i Sverige och i flesta större delen av västvärlden. Immunonkologispåret kom för ungefär 10 år sedan, kom första produkterna och Nobelpriset fick man för immunonkologiska konceptet 2018 tror jag det var. Där man hittade sätt att aktivera immunsystemet mot cancern. Normalt sett är cancer som växer okontrollerat, de är väldigt bra på att hämma immunsystemet. De skickar ut signaler som gör att immunsystemet håller sig i schack och inte stöter bort cancercellerna. Vilket faktiskt händer ganska ofta i oss, sannolikt att det bildas en cancercell som är farlig. Men den känns igen som kroppsfrämmande och stöts bort av immunsystemet. Och många framgångsrika cancrar har då haft den möjligheten att hämma immunsystemet. Och då kom Keytruda, Opdivo och Yervoy efter varandra för ett antal år sedan och tagit jättestor del av marknaden där man då kan aktivera immunsystemet mot cancer. Och det här är antikroppar som man har utvecklat som fungerar bra, men 30-40% av patienterna svarar inte på det här eller de blir toleranta mot det som de har effekt av mediciner från början men sen försvinner effekten och så kommer cancern tillbaka. Och det är jättestora marknader vi talar om potentiellt för Annexin. Om Annexin kan komma efter de här första antikroppen och sen kan vi ta hand om de andra potentiellt och skulle vi lyckas där så har vi 10 miljarder plus dollar i potentiella intäkter. Varför är vi då intresserade av cancer och Annexin? Jo, det är enkelt så att många cancerceller har man tittat på och de uttrycker PS, det vill säga har PS på sin yta. Och PS signalerar till immunsystemet, håll dig i schack, hämmar immuncellerna. Vi har inte gjort de här sakerna själva, studerat det själva, utan det är oberoende forskare som har studerat Annexin A5s effekter och pekade direkt ut det som en immunonkologisk checkpoint inhibitor. Till skillnad i samma effekt men separerat från de här storkännande läkemedlen. Tittar man på humana cancer som är farliga, det vill säga där patienterna har dålig överlevnad som exempelvis äggstockscancer, bröstcancer, mag-tarmcancer, bukspottkörtelcancer och även melanom, så finns det en koppling mellan hur allvarlig sjukdomen är och hur mycket PS som finns på cellernas yta, cancercellernas yta. Och det vill säga indirekt, potentiellt är det så att skulle man hämma PS, blockera PS med det här locket som Annexin kan lägga på PS, skulle man kanske kunna behandla många av de här svåra cancerformerna. Djurmodellerna ska jag inte gå in på i detalj. Det är några exempel bara där man stoppar in då humana cancerceller i möss och studerar effekten av olika läkemedel. Till vänster ser ni några olika linjer. Den nedersta linjen är då djur som dör efter 16-17 dagar. Då är 50% av djuren döda. Behandlar man med dagens mediciner så ser ni linjen i mitten, näst längst ner. Det är PD1 och PD11-hämmarna, CTL4. Sen kommer Annexin och sen kommer kombinationen av dagens immunläkemedel och Annexin och då dör inga av de här djuren alls. Liknande effekter ser man i en annan studie från Johns Hopkins University i USA där man ser att Annexin i kombination med ett cancervaccin fungerar väldigt bra och till och med bättre än dagens onkologiska läkemedel. Den andra möjligheten som vi ser framför oss det är cancer drug targeting som är också baserad på att PS finns på ytan av många cancerceller. Det finns koncept som finns idag som är stora marknadsledande produkter som har också en antikropp kopplat till ett cellgift som i sin tur binder till en specifik receptor på en cancercell. Principen är då att proteinet i dagens fall är då en antikropp som bär med sig ett cellgift, binder specifikt till cancercellen som har den här receptorn och tar cellgiftet upp och cellen dör så att säga, inifrån genom att specifikt rikta den mot cancercellen. Och det här är väldigt stora och viktiga produkter i dagens marknad. Annexin då, ja, samma princip skulle kunna gälla på alla de tumörer som inte har de receptorerna som det finns läkemedel för idag. Det vill säga, många patienter har PS-uttryckande cancerceller, är hypotesen. Vi sätter tillsammans med Annexinet och AnnexV en cellgift och så ger vi det till patienter så småningom. Hoppas att den kan avdöda cancercellerna men inte ge de här kända biverkningarna av cellgifter som håravfall, mag-tarmproblem, blodbrist av olika slag och infektionskänslighet. Och det håller vi på att arbeta med. Så det vi gör nu aktivt i cancerområdet är att vi har tagit fram ett protokoll för en klinisk utvecklingsplan inom cancer. Första studien är redan designad. Vi har förankrat den hos experter runt om i världen, Kanada, USA, Europa. Den är bra och kunna hålla för att gå in i patient så småningom, eller ganska snart om vi får godkännande och har finansiering för det. Och sen har vi då tagit fram en syntesmetod för att koppla cellgifter till ANXV som vi nu testar. Och vi förstås träffar också och pratar med stora läkemedelsbolag och mindre också och ser om de är intresserade av vårt cancerspår. Och sammanfattningens sista bilden är då en bild på vad vi gör under 23. Vi driver vår RVO-studie i USA, vi håller på med imaging-studien fortfarande, utvecklar cancerspåret. Vi pratar med många partners och hoppas att kunna 2024 starta fas 2B-studie i RVO och komma igång med cancerstudier i människa och i djur. Tack så mycket för att ni lyssnade på presentationen.
Och du nämnde ju här att ni rapporterade positiva första data i den här studien med patienter med RVO. Så jag är lite nyfiken på vad innebär det för utvecklingen av ANXV?
Ja, det kan man säga är hypotesbekräftande. Det vill säga att patienten med RVO har ett upptag av ANXV i ögat på de ställen där det ska tas upp. Så hoppas vi på att få in fler patienter. Det här var ett första exempel så vi behöver bekräfta det här. Vi kommer att kunna ge högre doser så småningom av det här fluorescerande ANXV. Och titta på, är det så att det binds mer vid högre doser? Alla patienter är lite olika, alla har inte samma typ av blockad i exakt samma ven utan det varierar lite grann. Finns det vissa patienter som har ett bättre upptag i ögat än andra patienter och det kan styra potentiellt urval av patienter så småningom.
Och sen ganska nyligen så riktar ni ju in er mot cancerområdet med ANXV. Så vad är nästa stora steg där för att komma närmare marknaden?
Ja, man får ju kommentera också varför vi som litet bolag väljer att gå in i ett så stort och för oss ganska okänt fält. Och det är mycket av det skälet att mekanistiskt sett så är de oberoende av varandra. Alltså om det fungerar eller inte fungerar i RVO har min uppfattning ingen bäring på om det kan fungera i cancer eller inte. Utan det är helt olika. Det är röda blodkroppar i ena fallet, inflammation i ögat. I andra fallet så är det cancerceller som uttrycker PS på ytan. Så de är oberoende av varandra och det är klart att det är ett stort steg men det är också en jättemarknad. Det finns ett enormt medicinskt behov. Återigen, även om det har kommit nya läkemedel, det kommer cellterapier i olika slag, genterapier som är jättepositiva så är det fortfarande den näst vanligaste dödsorsaken. Så att det är jag kan inte säga att det är ett moraliskt ställningstagande, för det är det inte. Men det är ändå en jättemarknad som är så viktig att bearbeta. Så det är en stor steg för bolaget. Men vi arbetar med i princip har en kärngrupp av journalister i bolaget kan man säga. Vi har Anna då som är specialist på PS och Annexin förstås då. Men sen har vi då experter som jobbar med det. Så det är en effektiv arbetsmodell som man kan tillämpa tycker jag.
Mm. Och avslutningsvis ska du få berätta vilka milstolpar du hoppas på att uppnå här innan årets slut.
Ja, vi hoppas förstås att kunna presentera säkerhetsdata från den första patientgruppen 8 patienter i RVO så snart som möjligt. Vi arbetar ständigt och till och med senare idag på eftermiddagen kommer vi att prata med våra läkare i USA om dela erfarenheter för att få dem att vara mer entusiastiska och rekrytera snabbare och bättre på alla möjliga sätt. Så hoppas vi vara klara med studien som helhet i slutet av året. Så vi har data vi kan presentera. Vi hoppas kunna hitta en partner. Vi kan diskutera med många olika företag. Allt sånt där är väldigt hypotetiskt, man kan inte lova någonting. Men det är i alla fall intressant att det finns ett intresse för oss. För vår produkt och förstås. Den är så annorlunda än anti-VEGF. Alltså det kräver lite övertalning och kanske lite förståelse för vad vi egentligen vill åstadkomma. För de ska förstå, för det domineras helt och hållet av injektioner i ögat och andra typer av läkemedel. Inte minst anti-VEGF för de nya sorter. Sen hoppas vi kunna presentera att vi har lyckats döda cancerceller i labbskålar. Där vi kan döda dem bättre eller mer effektivt än dagens sådana här konjugat cellgiftkopplade läkemedel. Kanske kan vi hitta effekter i djur också innan året är slut.
Okej, ja, det är mycket att hålla koll på där helt enkelt. Det är det. Tack så mycket för att du kom hit.
Tack så mycket.
Då är det dags för en presentation av Ziccum och VD Ann Gidner. Varsågoda.
Tack så mycket. Jag är mycket glad att vara här och presentera Ziccum idag. Vi torkar biologiska läkemedel med framgång eftersom vi gör det med masstransport. Först har vi en liten slide här bara om framåtblickande uttalanden. Men sen vill jag direkt gå in på 3 nyheter som vi levererar. Jag är väldigt glad att kunna presentera det här idag. För det första så har vi framgångsrikt torkat mRNA i LNP-partiklar. Vi har också bevisat att mRNA behåller sin aktivitet. Jag kommer in mer på detaljerna här. Men jag kan säga att det här är den största tekniska milstolpen för Ziccum hittills i bolagets historia. 20 mars kunde vi gå ut med det. Vidare så har vi gått in med 3 stycken patentansökningar. Vi jobbar med en ny strategi för att skydda IP, Intellectual Property, i många lager. Vi har 3 patentansökningar registrerade 28 april. Den första gäller utrustning där vi kompletterar det befintliga utrustningspatentet Ziccum har. Det andra gäller formulering av läkemedel eftersom vi har vår egen specialformulering för att lyckas med den här speciella torkmetoden. Den tredje ansökan gäller processparametrar där vi ser till att köra vår process optimalt för att få bästa resultat. Och sen har vi då i maj en tredje nyhet som är den allra största och det är att vi skriver ett avtal med en ledande mRNA-tillverkare, mRNA-spelare på den globala läkemedelsmarknaden. Vi skrev det här 9 maj och kunde gå ut med det. Det är också viktigt att poängtera att det här är vår första kommersiella intäkt i Ziccum. Så kommersiellt sett är det här en väldigt stor milstolpe för bolaget. Då tänkte jag gå in lite mer på teknologin, laminate-paste, som vi arbetar med. Ziccum utvecklar laminate-paste. Det här är en teknologi för att på ett lyckat sätt torka biologiska läkemedel, för det är väldigt svårt att göra det. Den här innovationen gjordes ursprungligen för inhalation, så den fungerar utmärkt för inhalerade läkemedel. Det är en unik torkmetod eftersom den bygger på masstransport och därmed kan ske i rumstemperatur. Så vi behöver varken frysa eller elda upp materialet. Vi kan ha en väldigt skonsam metod och det är det som gör en unik process. I nuläget har vi en enhet som vi utvecklar för industriell implementering. Just nu är det 4:e generationens pilotanläggning vi har på bolaget och vi har ett stort projekt där vi tar den här till färdig GMP-version. Och vi kan alltså torka biologiska läkemedel på ett lyckat sätt. Vi har använt utrustningen och kört provförsök för en lång rad substanser som används som biologiska läkemedel. Det är protein, det är peptider, det är enzym och antikroppar. Och det är också vaccin på 3 olika plattformar som vi fokuserar på. Vi har lyckats torka många sorters substanser men vi fokuserar strategiskt nu på vaccin på 3 plattformar. Framförallt de 2 sista här, det är protein subunits och det är mRNA i LNP-partiklar. Och för att då komma in på mRNA lite mer så har vi då 20 mars gått ut med resultaten efter en lång rad försök av både modellmolekyler och äkta mRNA i lipida nanopartiklar. Och det är de här väldigt komplicerade strukturerna som syns på bilden här, där man har en stor mängd fettsyror, lipider, som bygger upp en sfär och bär med sig mRNA. Det är det här som har möjliggjort de nya covid-vaccinerna. Tack vare LNP-partiklarna så kan man få mRNA att fungera in i människokroppen. Våra resultat visar att vi behåller stabiliteten mycket väl, för det här är en väldigt känslig struktur. De här partiklarna kollapsar ju eller aggregerar, klumpar ihop sig väldigt lätt. Men vi kan behålla rätt struktur. Vi behåller också tillräckligt mycket mRNA. Mer än 95% finns kvar inne i vesiklarna i strukturen. Och vi har också en perfekt storleksfördelning på partiklarna. Andra metoder där man försöker hantera biologiska läkemedel, där blir det lätt aggregering och alldeles för stora partiklar. Sist men inte minst så har vi bevisat mRNA-aktiviteten. Med in vitro-försök i vårt eget cellabb så ser vi att mRNA är lika aktivt, så att det har helt kommersiellt gångbara nivåer, vilket också är unikt. En liten titt då på varför. Varför vill man göra om flytande biologiska läkemedel till torra pulver? Då är det ju så att, som säkert varenda människa känner till idag, att covid-pandemin skapade ett enormt behov av nya vacciner. mRNA-tekniken var framme och färdig att användas, men behövde levereras in i människan. Det gick tack vare LNP-strukturerna. Men eftersom de är så komplicerade och känsliga så kräver de hittills förvaring vid kryogena temperaturer, minus 80 grader. Och eftersom hela världen var så angelägna om att få fram vaccin så accepterade man det här problemet. Det kostar mycket tid, mycket pengar att hantera produkter ända ut till varenda användare i den här fryskedjan. Men för att få vaccineringen lyckades gjorde man det. Men med vår teknologi skulle man alltså kunna få en oändligt mycket enklare process om man då får bort kylkedja och fryskedja ut till användaren. När man gör det här så blir det en enorm vinst också i hållbarhet, sustainability. Tittar man på koldioxidavtrycket för vaccinbehandlingar så är över 90% beroende på kylhantering och fryshantering. Så att med en enkel pulverhantering istället har man en mycket stor vinst vad gäller hållbarhet. Och för det tredje så kan man ju då också få helt nya administrationsvägar till patienter. Man behöver inte gå tillbaka till vätska och ha komplicerade injektioner. Man kan till exempel inhalera pulvret i sin existerande form. En snabb titt på hur konkurrentsituationen ser ut, hur marknaden för att torka läkemedel ser ut. Där är den gamla metoden, frystorkning. Det är en väldigt low-tech, gammalmodig modell, simpla brickor in i ett frysskåp och svårt att kontrollera, svårt att hålla läkemedelsstandard. Industrin har gått vidare till spraytorkning. Det fungerar väldigt väl för små kemiska aktiva substanser men inte för biologiska läkemedel. Det blir många steg, en ganska så komplicerad process och den utsätter produkter för både värmestress och mekanisk stress, vilket inte fungerar för biologiska läkemedel. Och därav att det skapar ett sådant intresse i branschen att vi har en helt ny metod för att torka biologiska läkemedel. Det kan nämnas här också att det är inte bara så att vi har ny teknologi med alla dess möjligheter. Samtidigt så har vi högt utbyte och en enstegsprocess. Så vi tror också att det här kan implementeras utan stora kostnader. Det kan bli mer kostnadseffektivt än befintliga metoder. Sen kommer några ord här om marknaden. Vaccinmarknaden har ju förändrats totalt tack vare, kan man säga, pandemin, covid-pandemin. Alla de spelare som tidigare var vaccinutvecklade bolag vill ha ny teknologi. De gör allt för att komma åt ny teknologi. Bolag som inte tidigare jobbade med vaccin vill in i marknaden. Siffrorna här visar de 3 vaccinplattformar där vi är aktiva. Och om man räknar på en royalty-ström på 1% av vaccinförsäljning för våra 3 plattformar och man tittar bara i västvärlden, då motsvarar det 400 till 500 miljoner dollar per år i marknadspotential. Ett annat sätt att titta på den här marknadsmöjligheten det är mRNA. För mRNA kan vara mycket mer än vaccin. Så att vi hade en del av staplarna i förra bilden var mRNA-vaccin. Här är det istället alla tänkbara mRNA-projekt som visas. Det finns cancerterapi, det finns personalized medicine, det finns en lång rad projekt i klinik och i preklinik. Och man förutspår att det här kommer att närmast explodera. Jag har ett citat här. Med bättre utvecklad patientadministration och stabilitet så kommer mRNA-plattformen att ge potential till en lång rad nya behandlingar och terapier. Och det här är ett citat vi gillar väldigt väl eftersom vi bidrar till en bättre patientadministration och vi kan helt lösa stabilitetsproblemet. Så att Ziccums teknologi är ju en väldigt viktig faktor i att åstadkomma den här förändringen. Jag vill också säga här att det är inte bara så att vi har en enorm marknadspotential långt borta när en produkt väl når marknaden. Det är också så att på grund av de här stora möjligheterna, möjligheten till helt nya patientbehandlingar, möjligheten att lösa svåra vaccinproblem med mRNA, gör att det investeras som aldrig förr. Så att här och nu idag så sker ju historiskt höga nivåer av investeringar i läkemedelsbranschen för att så många spelare vill satsa på mRNA. Så att här på bilden är 6 olika exempel från $10 miljoner till $270 miljoner på stora satsningar, partnerskap, investeringar, och då är det här bara industriella satsningar. Vi har inte med alla statliga satsningar som också görs. Och med det sagt så är det ju fantastiskt att vi har nyheten för Ziccums del att vi nu har skrivit på ett utvärderingsavtal med en ledande mRNA-spelare, en globalt ledande spelare som vill utvärdera Laminar Pace tillsammans med oss, dels för mRNA-LNP-material och eller mRNA-vaccin. Det här skrivs på 9 maj. Det är en fullt betald studie så att vi får in flera miljoner på det här projektet. Det är också Ziccums första kommersiella intäkt, så vi är enormt glada för det här. Vi startade projektet igår och ser verkligen fram emot det här samarbetet. Det är så att det är inte bara så att vi tjänar pengar, det är också så att vi lär oss enormt mycket när vi får komma ända in i hjärtat av mRNA-utveckling hos de här storspelarna. Så vi lär oss ju fantastiskt mycket på resans gång. Vi är också i fler dialoger med fler spelare, så vi hoppas ju kunna presentera nyheter fortlöpande. Och det kan också sägas att med den här intäkten så behöver vi inte oroa oss för finansiering i närtid. Och till sist, bolaget Ziccum, vi finns i Lund och vi är noterade på Nasdaq First North. Och med det vill jag tacka så mycket. Tack!
Ann, jag tänkte att jag börjar där du slutade med det här viktiga avtalet. Skulle du kunna berätta lite mer om hur det ser ut ekonomiskt?
Absolut. Det är ju väldigt roligt för oss eftersom det är vår första kommersiella intäkt. Så att vi har ett avtal där vi tar in allt arbete betalat. Vi tar in flera miljoner till Ziccum. Vi har också en del förskottsbetalning. Så att det känns som en bra deal för oss. Och dessutom gör det ju då att vi säkrar upp så vi inte behöver finansiering just nu i en ganska svår marknad.
Och som du också nämnde i presentationen så har ni kunnat leverera ganska tunga nyheter här, slag i slag ett tag under våren.
Ja.
Skulle du säga att det är ett resultat av den här nya strategin som ni införde?
Jo, men det är det absolut. Det är en rad faktorer. En ny strategi kom in förra våren. Jag kom in som ny vd i maj. Sen är vi omorganiserat. Vi har lagt en ny affärsmodell med just betalda lukrativa studier så att säga. Vi har också säkrat upp mycket mer strukturerade försökskörningar för egen del. Vi har ritat upp ett knowledge space och ett design space för vår utrustningsperformance så att vi har mycket bättre kontroll på försöken vi genomför också. Och det har ju lett fram till de här fina mRNA-resultaten, vilket ger intresse, vilket hjälper affärsutvecklingen. Så att det är en positiv spiral kan man säga.
Ja. Och om vi slutar med att blicka framåt lite, vad händer näst då i den här positiva spiralen?
Vi fortsätter nu, vi vill ju arbeta. Jag vill arbeta väldigt aktivt. Jag har ett stort nätverk inom pharma. Vaccinindustrin är verkligen på tårna just nu och vill ha ny teknologi. Och vi har goda resultat. Så att vi kommer att driva, dels kommer vi ju förhoppningsvis att ta befintliga samarbeten till ett nästa steg. Vi kommer också starta fler dialoger. Vi har ett antal dialoger på gång. Så att vi jobbar parallellt med flera steg. Vad gäller egna försök så har vi kommit fram till väldigt bra resultat. Så vi kommer satsa mer på partnerskapsarbeten just nu.
Men då ser vi fram emot fortsatta nyheter från Ziccum.
Ja tack, det ska vi göra.
Och tack så mycket för att du kom med.
Ja, tack så mycket.
Next up is Karin Brider from Colzyx. Welcome, Karin.
Thank you. Antibiotic resistance. That is a huge problem in the world today. It affects us all by a growing number of infections where antibiotic doesn't work anymore. This is a great burden for the society and also for the patients, of course, that are affected. Actually, in 2019, almost 1.3 million people died from bacterial infections with strains that were antibiotic resistant. So, now I would like you to join me on a mission to fight antibiotic resistance. We have a peptide technology that actually has the power to reduce the use of antibiotics. And why is that important? Well, the main driver for resistant development is actually the use of antibiotics and an overuse of that. Our peptides also destroys already existent resistant bacterial strains, and on this picture the arrow shows all the strains that we've tested, and they all are killed by our peptides. We have a second mission that I also would like you to join us in, and that is to improve wound healing, and especially for hard-to-heal wounds. Hard-to-heal wounds, well, the most known one is, of course, diabetic foot ulcers. But there are also pressure and leg ulcers in that category. Most, if not every, hard-to-heal wound are infected with bacteria. They're dormant, but sometimes they get activated and then you have a huge infection that is almost always treated with antibiotics. So to be able to heal a hard-to-heal wound, you need to attack the bacterial infection as well as making a milieu for the tissue to grow back. So at Colzyx, we develop bioactive products, and we're using peptides from collagen 6, Colzyx. Collagen 6 is present all over the body, together with its family members, collagen 1, 2, 3, and so forth. Interestingly enough, collagen 6 is upregulated when you get a bacterial infection, example in the skin. And the molecule is then degraded into small peptides that are active within the wound. So our first product, WoundCom. WoundCom is a medical device. It's a wound dressing. It's built from collagen 6, a collagen 1 matrix, and then added on with collagen 6 peptides. As you can see on this picture, the WoundCom that is situated in here in the middle in a wound is very like the dermis, which is this one. It's the low part of your skin. Magnifying this, you can see closer into WoundCom, and it has a very structural appearance with pore sizes that is well known to amplify cell proliferative activities. Then one magnify this even further and visualize our peptide with immunostaining. You can see that the peptides are evenly distributed throughout the product. So if one then takes this material and put bacteria on it, on the lower two panels, that is pure collagen 1, a matrix, the matrix, and then the upper two panels are WoundCom. The green are bacteria, and the pinkish thing is dead bacteria and debris from bacteria that is actually sticks to the collagen 1 matrix when they burst. As you can see, having the peptides in WoundCom enhances the effect, the antimicrobial effect extensively. On the other hand, if putting human cells on the matrix, you can see that the purple ones here is cells from skin, and adding that to collagen
makes them grow very well. So in a couple of days, they multiply. Having our peptides in WoundCom augments this activity even further. So in real life, we made a porcine wound model where we inflicted wounds on healthy pigs, infected them heavily with Pseudomonas, and then treated with either a normal wound dressing, like a natural healing situation, the pure collagen dressing, or our WoundCom. A normal wound healing in a healthy pig takes about 3 weeks. With a collagen wound dressing, it takes about 2, and with WoundCom, the wound were healed within the week. So, in summary, WoundCom contains of natural occurring molecules that are completely compatible to the body. We do decrease the need for antibiotics because we kill the bacteria in the wounds and it heals faster. The project is now in clinical investigation phase. We're awaiting green light for our proposed clinical investigation. We believe that WoundCom is well situated in the landscape of wound dressings. We attack both the antimicrobial properties and the wound healing promotion. The market. So the market size today for collagen wound dressings is $2 million. The biggest players are, of course, the US and the Middle East. And it's noted that the collagen wound dressing is just 10% on the overall advanced wound dressing market. We believe that WoundCom will have an addressable market that can take all advanced wound dressing properties. With that said, our first target is hard-to-heal wounds. In that target group, you have a prevalence of 2 million patients globally. And that is rising because we get older and we get sicker. So this will even be further enhanced along the years. Our second target is, of course, that we would like to expand to acute wounds. So after the pandemic, we'd had some interesting discussions with future distributors. But of course, in this case, the next step is awaiting clinical investigation. Interestingly enough, we've had interest from veterinarians that have tested the product and found it quite good. So that is maybe an indication of that our first market would be animal care. I've been talking a lot about WoundCom, but we have a peptide platform, and we've had some interesting discussion for licensing agreements for partners that then will use them in their specific development programs. So to us, we started the company in 2016 and got our first bag of money in 2019. Today, we're 4 very dedicated people working with the project and the company, and we are fully equipped to take this company and the project we have one step further. So as we have grown from 2016 and made a lot of progress in our product, we've been funded by 68 million Swedish kronor. And we've had the fortune to be partly funded the WoundCom project from the European Union Horizon 2020. And as you can see, the distribution of shareholders are fairly equal between the founders and external investors. So, of course, now we're here and presenting, and we're looking to raise new capital. We would like to be able to have some resources into the distribution into the animal market. And also, as you saw, the the big player is US, where collagen especially and advanced wound dressings are accepted. We would like then to be able to put in an application for an FDA approval. Why we're not doing that in the first case? Yes, we got the EU grant and we're of course obligated to look into the EU market. Going further and having maybe the cap, which at 40 million, we would like to be able to expand our user claims of WoundCom. That requires additional clinical investigation, and that, of course, as you know, costs a lot of money. Of course, also we would like to investigate our fully platform. We're now focusing on 2 peptides. We have a set of 20, and we would like to make feasibility studies and to additionally use of the whole technology. So I hope I've convinced you to join me on this exciting journey. And of course, you're welcome to contact, and I would like you to contact me or Jens for further questions and some discussions. So, as I said, thank you and hope you join me.
Where do you see the company in 2 years?
So the company will have some license agreement and some development programs with the peptides, with other peptides or the same peptides that we use in WoundCom today. The WoundCom project will be in the phase of either having a CE mark or actually in the end of receiving a CE mark, because we have the upcoming trial or investigation, clinical investigation first, and then we have to apply for a CE mark. And unfortunately, the notified body system, as you know, now is kind of struggling with the new regulations, and it takes a lot of time. This is also a very complicated product. But for the other ones, and I think we're having a distributor for the animal care. We have, if I can dream, we actually have an FDA approved, and maybe have made a study in the US to be able to then market it. But in 2 years, I think it's too early to say that we're having this great market access, but still we're kind of on the border to actually go in there.
And what does the competition look like in this field?
So the competition is, of course, a lot of advanced wound dressings. If we look at the wound dressings, and there are different peptides coming up here. We have some studies in Sweden, and they're also looking at the same thing as we. We have the collagen market that is quite extensive, and a lot of products are really good, but none of them really have the dual activity that I was talking about. So it's also that, of course, we need a lot of proof before anyone will use it and buy it. So it is a huge market. It is a lot of competition. I do think some of them, because of the new regulation, will actually go away. And the use of silver and some other chemicals that have been kind of looked into that are cancerogenic, you have to stop using those. So I think that's kind of where we are. With upcoming projects, there are some upcoming projects, and we'll have to see where those leads, of course, if we're kind of in the same area.
Thank you, Karin, for joining us today in the studio.
Thank you.
Next up is Torbjörn Bjerke from SynAct Pharma. Welcome.
Thank you very much, and thank you very much to BioStock for inviting us for this spring summit in life science. So I'm Torbjörn Bjerke. I'm the CEO of SynAct Pharma, and what I'm going to talk about today is what is important for SynAct Pharma during this year. And as many of you know, we are focused on treating inflammatory diseases with a totally new concept of therapy, that is resolution therapy. And I will come back to that as well. So, just some forward-looking statements. And then let's just take a little bit view on what happened in 22, which was quite an important year for us. And obviously the uplisting from Arcede to Nasdaq Stockholm main market in the mid-cap section was important for us. And thank you to Patrick Renwald, our CFO, for that. It was really important for us in order to capture more investors. And then importantly also was the okay from the American authorities, FDA, for us to start clinical development in the US. This is a really a quality statement on our programs. And thank you very much to Thomas and Mette and the others in the R&D department for that. Importantly also was the initiation of the ongoing clinical trials in RA as well as in the kidney space. What I will focus on today is really what's going on in the RA field. So let's move on to that. But first of all, I just want to give you a snapshot of the pipeline. This is obviously the value of SynAct Pharma, all the different product opportunities within both large indication areas, but also some more narrow indication areas. And that is what is a good quality sample of a good pipeline. So as you can see here in the first two columns is all about the rheumatoid arthritis in two different patient populations. I'll get back to that. And then the nephrotic syndrome. In addition to the virus-induced respiratory insufficiency, that is our second proof of concept that our lead molecule, AP1189, works in inflammation. As some of you might remember, we got really good results from the COVID-19 induced pulmonary inflammation, and in addition to the RA. And then following the acquisition of GXP Pharma earlier this year, we are adding on some really exciting opportunities in other spaces as well. And one of them is the prevention of organ failure in complicated surgery. So I would say a very good pipeline. So it's all about inflammatory diseases. And when you have an inflammation and you cannot deal with the inflammation, it can be chronic, become chronic. It's an evil way of the inflammatory response to just accelerate. It's important for us to stop that building up into a chronic inflammation. And our compound 1189 works in two different ways. One is to slow down the inflammatory response by inhibiting some of the different mediators and cells that are important in the inflammatory response. That's one thing. And the other thing is to clean up the mess. So the compound also stimulates the so-called, I call them trash cells. These are the cells that are cleaning up the debris from the inflammation. By doing those two things at the same time, we believe you can rebalance the immune system. You're not inhibiting one substance that many of the treatments in RA does and causing immunodepression. What we are doing is rebalancing. We believe it's a much more intelligent way of stopping the inflammation. Why rheumatoid arthritis? I often get that question. It's a very, very big area. It's about 1% of the whole population in the world. And about 70-80% of those that suffers are women. The reason is, yes, it's big. However, more importantly, it's a big medical need. Even with quite a lot of different treatment options today, you don't get full control over your symptoms. And that's important. So it's really room for improvements here. About 50% never gets this full recovery of the symptoms. And this is a very serious disease that are causing a lot of troubles for those that suffers from it. So that's why. This is a slide showing some of the important results from the first study we did in these patients. These were newly diagnosed, very severe patients. So they were treated with methotrexate. That's the normal treatment that you get when you are diagnosed with the disease. And then we added 1189 in 2 different doses, 50 mg per day and 100 mg per day. And then the grey one there is the placebo, and that's only methotrexate. And as you can see here in 2 different very important readouts, first the one is the C-line score, the CDAI. And what you can see here from the baseline is that the dark blue has a significant effect. So 100 mg had a significant effect on that. The other one was the ACR 20, 50 and 70 response. This is the normal regulatory response from the authorities in the US, the FDA. They require ACR 20 being significant. And please remember that this is only after 4 weeks of treatment in a chronic disease. Here we also got, in the ACR 20, we got a significant response. So these 2 readouts are extremely important in RA, and we got a significance on both already after 4 weeks. 2 other readouts. One of them were the so-called DAS28. Here we include the acute phase protein, a biomarker in the blood called CRP, which comes if you have a chronic disease in some of the patients. And here we got significance as well. And as you can see on all of these results, you can see a dose response, which is very important in drug development. Lastly, but not least, we got a good change in the fatigue score. Many of those patients suffers from very severe fatigue, and this is a devastating symptom that they do have, and we got a good response there as well. All of them being quite clinically significant, which is of course also important. But remember, again, this is already after 4 weeks. So another thing was to look at the safety in the trial. And most of the drugs that are used today in RA do have a lot of safety problems because they are very much taking out the immune system or inhibiting it in a very high degree. So safety is important. And as you can see from this slide, our safety profile from the beginning was very benign. A good safety profile. No serious adverse events in this trial here. And the adverse events that we saw was mild to moderate, no severe, except for 2 in the placebo group. So a good safety profile, important. So what we're doing now, and what you should be looking for in the second half of this year, are readouts of two trials. And the first one is the so-called EXPAND study. This is an expansion of the BEGIN, exactly the same group of patients, very severe, and we're treating for 12 weeks now, 12 weeks. And we have ACR 20, as you remember, the regulatory readout from the FDA, American authorities. We have that as a primary readout. But you're also looking at quite a few other things. in this trial. So it's a bit copy-paste to begin, however, with 12 weeks to see efficacy after 12 weeks, to see safety after 12 weeks. So that's important. Another important trial is the proof of concept in another group of patients. These are the patients that have gone through methotrexate treatment, however, is still having symptoms. And then we add on 1189. And we treat for 4 weeks on the part A of this in different dosing. And what we're looking for here is to see if we can see an effect on these phenotype of patients. This is quite an important study as well, reading out in the second half of this year. So the emerging profile of the 1189 is really, really exciting. Once daily oral dosing, a quick onset to action. We have seen that in both trials that we have done. So this is important for the patients, obviously. High degree of efficacy. We can see that from both the BEGIN study as well as the COVID-19 study. Safe and well tolerated, which is important, and so on and so forth. So this profile can be put into the treatment concept of RA. And so far, we can say that, yes, both the quite severe patients, newly diagnosed that needs treatment, can be a good target for this and a very big market. But even a bigger market is the so-called DMARC IR, which is the phase A of the RESOLVE study that we are doing this year. This is just to say that the newly diagnosed, a big group of patients with big medical need. We have asked different key opinion leaders in the US about the profile so far. Take-home message here is that we would love to use it. Obviously, we need more results in order to substantiate the profile, but so far it's really, really promising. Several new indications. My time is running out, so several new indications. We can use this we believe as a general inhibitor of inflammatory diseases. We see some of them on this slide here. Big market opportunities, big medical needs as well. Thank you.
Where do you see the company in 2 years?
Where I see the company in 2 years? I see us being even more established within inflammatory diseases. I see us really building that franchise within, as I talked about a little bit today, about the resolution of inflammation. I think, I've been in the pharmaceutical industry for 30 years. I see this as an extremely exciting new field where we can go in and educate the immune system to rebalance the disease. So I see us being even more established within that field. I see us also having a rather big partnership because what is important for 1189, as I see it, is to have enough power to go, and I mean financial power, to go into quite a few different indications. Because, as I said, this could potentially be used quite broadly in inflammatory diseases. So even more established, we have a good pipeline, many optionalities in that, but I also see us having a partnership.
But what does the competition look like in resolution?
The competition in resolution, now we are focusing on melanocortin receptors, and there's not a lot of competition there. There are some in the US, but they are using some other means in order to for example, MC4 receptor has been used to treat obesity, especially in more seldom diseases within obesity, with great success. So there's a company called Medicon that have done that very successfully on the Nasdaq Stock Exchange. There are also some other attempts to do resolution therapy. However, they're not that as advanced as we are.
Thank you, Torbjörn, for joining us today in the studio.
Thank you very much.
The next presenter is Lars Gusch, CEO at Pharmacolog. Welcome.
Thank you very much, Nicola. So today I will speak about medication safety and drug diversion prevention, as suggested by the headline. You may ask yourself why. Yeah, because it's a problem if you don't address those two things in your daily use in medication practice. So you may or may not know, but here are some facts about the price of the errors in medication safety and drug diversion prevention. There is actually an increased pressure on pharmacy personnel, which leads to medication errors. And that causes patient harm, material damage, and worst of all, patient deaths. If you look at the middle of the slide, you see the price of the errors. That is 1.5 million Americans are harmed by preventable medication errors annually, and this causes a damage of about $3.5 billion per year. If we look at drug diversion, that's about the same thing. We see drug diversion as a part of the opioid crisis in the US, and this causes also suffering for people, material damage, and fatalities. Avoiding those problems is what we work at Pharmacolog every day. So my name is Lars Gusch. I'm the CEO of Pharmacolog, and I want to tell you a little bit more about our company, about our technology, about our solutions, and how we want to conquer the market within medication safety and drug diversion prevention as Pharmacolog. The company was founded as a medtech startup in 2007. Our first commercial introduction of the drug log product was in March 2017. We introduced another product in 2020, which is called WasteLog, and both are systems for verification of injectable medication. We are listed on Nasdaq First North, and we are present in more than 45 hospitals throughout the whole world, but mainly in the US and Europe. And there are big hospitals that has chosen our solutions. For instance, New York Presbyterian, UCLA, Johns Hopkins, and UT Southwestern. And the most important of all, with our installations of about 107 devices, we have a database now of more than 150 identifiable substances, which covers 80 to 90% of the most common substances to be analyzed in those hospitals. So how does our technology work? It's based on absorption spectroscopy. It's a rather classic technique. It has been there since the 1700s. But we actually put a special touch on that technology by analyzing the broadband spectrum of the substances. And this gives us our unique IP and also gives us the identification of both the substance and the concentration of the substances. It's also worth to mention that our technology requires very small sample sizes and is non-destructive. So what else makes us unique? Yeah, it's the broadband spectrum analysis, which I mentioned before, but it's also our advanced data processing and analytics, our IoT capability of our devices, which actually, in a combination, gives us big data in our field. So we have an enormous number of measurements and analysis from customers all over the world, which we can provide then back to other customers all over the world through our IoT capability of the devices. So let's look at where in the medication use process we position ourselves. It's actually in medication safety, where we do quality control, and it's also in drug diversion prevention, where we do waste screening. Our mission is to provide solutions and products within drug administration and delivery for accurate and individually optimized patient treatment. Let's deep dive into medication safety and quality control. So what is it about? It's about medication errors. As I mentioned before, drugs in the medication use process have to be delivered with the right drug and the right concentration. If that doesn't happen, then we have a medication error. So the WHO estimates that medication errors causes about 163,000 deaths in Europe alone, which corresponds to a cost of approximately 7 billion euros. And this affects all patients, relatives, and healthcare staff. And of course, by that has increased focus from the authorities, both in the United States and in the European Union. So guidelines are put in place which actually require quality control of the prepared substances. So we are going from QA to QC in the good manufacturing process of medications. And we are there with our solution, DrugLog. That targets a market of about 3,500 hospitals in Europe, globally about 15,000 hospitals, and has a market size of 100 million euros, 180 million euros. If we look at drug diversion prevention, it's actually the diversion of potent drugs, so basically the stealing of drugs within the hospital, that is the problem. It happens in all healthcare facilities, and very potent drugs are diverted or taken by the hospital staff, including fentanyl and hydromorphone, very potent drugs. And of course, this also leads to the focus from the authorities, because the opioid crisis in the US is a very big problem. There has been guidelines put in place, and the DEA is controlling that those guidelines are followed and drug diversion is reported. So 2/3 of professionals perceive risks of fines in their hospitals from the DEA, and we see just the beginning of implementing those drug diversion programs here. We are there with WasteLog as a key element in the chain of custody. The market potential is about 5,000 acute care hospitals as a primary target for our solution, but also with additional markets in ambulatory surgical centers and palliative care centers. The estimated market size is about $840 million US. And the competition is basically non-existing at the moment. It's a 4I methods, so basically witnessing from a human being when wasting drugs. So where are we now? We have actually established our solutions, DrugLog, in the European market, in Belgium, France, and Spain. And we have also established WasteLog as drug diversion prevention key element for very huge hospitals in the United States, like UCLA, New York Presbyterian, and UT Southwestern and Johns Hopkins hospitals. We have two very competent partners on hand in the fields of medication safety and drug diversion prevention with B. Braun in Europe and Codonix in the United States. But we also want to accelerate our business and want to make our products available quicker and to more customers. And this is our future strategy. We want to lowering the investment threshold for our systems by implementing a software as a service business model, basically split of the high capital investment and keeping the scalability both for Pharmacolog and our customers. We also want to look at our distribution channels. We want to broaden our distribution channels regionally
And we're looking into integration with medical information systems as a competent partner in the field of in this market segment. So as a summary, we think we have a very strong technology and IP. We have proven revenue and an established customer base, and by following the market and seeing the increased focus on both areas, we see a lot of growing market potential. And with our new strategy of accelerating the sales with the software as a service model and expanding our partnership and distribution network, we feel that we can increase our market very much in the future.
And my first question is about your latest report, because during Q1 this year, you achieved your highest sales report so far. Would you like to explain what is behind this increase?
Yeah, this is actually a result of our partner network with our partner Incodonics, mainly in the US. So they ordered a lot of systems now, which is the main reason for the increase. But we also see an increase of system sales in Europe for the quality controlling area.
I see. And you are new as a CEO of the company. What is your vision?
Well, my vision is to make this technology available to even more customers and actually focusing even more on those two areas, medication errors and drug diversion, to prevent those two things with our systems and integrate the system into the whole workflow of the medication use process.
You have a rights issue coming up in June, so I'm curious, how will you spend the money?
Well, we plan to spend the money actually on commercialization of our products. So by broadening our distributor network, broadening our sales efforts, and also build the system, implement the new strategy as software as a service, improving the products towards that, and also integrate with other partners there. So that will be the bulk amount of money that we plan to spend there.
I see. Good luck, and thank you so much for coming.
Thank you very much.
for our last presentation of the day, we have Arcede Pharma, a biotech developing drugs for the treatment of chronic respiratory diseases. Here to present the company is CEO Mia Lundblad. Welcome, Mia.
Thank you very much. So, Joe, during this coming session, I will present Arcede Pharma to you and give you a status of the current development project Arcede 405, but also give you the plans for going forward. So starting with the vision of Arcede Pharma, and that is to provide treatments of chronic obstructive airway diseases through new and innovative drug candidates with the aim of obtaining fewer and less severe symptoms, improved quality of life, as well as prolonged survival. And through this, we are also contributing to one of the specific goals on the UN Agenda 2030, which is to decrease the number of deaths from non-transmittable diseases. So coming back to the actual disease that we aim to treat, which is chronic obstructive pulmonary disease, COPD. It's a disease with a high disease burden and also with a high unmet medical need. Actually, it's the third leading cause of death globally, with more than 3 million people dying of the disease in the world per year. It's also a disease where available treatments actually have poor efficacy and also are associated with safety concerns. Available treatments also lose their effect over time, and they don't really target the underlying disease, but more the symptoms. And thus, there is an urgent need for new and innovative treatments that actually can have an effect on the exacerbations, that is, the acute worsening of the symptoms, and thus also prevent or at least stop the disease progression. Since actually, despite optimal treatment, more than 30% of patients still experience exacerbations, and about 40 to 50% remain symptomatic. It's not only a disease with a high disease burden for the individual patient, but the economic burden is also high on society. The actual healthcare costs amounts to about 40 billion euros in EU only per year. And as you can see from this graph here, the main contributing factor to these costs are the hospitalizations. And the hospitalizations are mainly caused by these exacerbations related to the underlying inflammation. So it's actually very important to be able to target the actual underlying inflammation. And here we see that Arcede 405 has a great potential. We have demonstrated anti-inflammatory effects previously in vitro, and now we have also confirmed these results in vivo. And as seen here in this graph, there is a decrease in the recruitment of important immune cells, such as the neutrophils and the inflammatory macrophages. And that is seen as the decrease to the left with the dark blue dots in each graph compared to the vehicle to the right in each graph with the light blue dots. And this decrease in immune cells also then causes an inhibition of important pro-inflammatory cytokines. Arcede 405, our drug candidate, not only has this anti-inflammatory effect, it also has an airway relaxing effect, which we previously have demonstrated in ex vivo studies, but now also in vivo studies. This is shown here in this graph as the reduction from the red line, where we see decrease in the resistance with treatment of Arcede 405 in the blue line. And this is apparent both for the lung constriction, but also for the peripheral and central airway constriction. And with these very positive results and with the beneficial properties of Arcede 405, we are of course very eager to continue our development with our compound. And this has now also been possible, and we can actually increase our pace here with the fact that we now have received a grant from Vinnova SweLife, together with Lund University, Novo, and ourselves, Arcede Pharma, as the project coordinator. So with this, we can focus our development activities a little bit more and at a more constant pace. And some of these activities that we intend to perform is actually to set the therapeutic concentrations and relate those concentrations to the potential side effects that we would see in the tox studies. And with these concentrations, we will be able to establish a therapeutic window so that we in the end can predict the clinical doses. Because the clinical doses are, of course, also very important to know for the formulation development and optimization that is being performed by Novo at the moment. Because they need to know the doses and amounts of RCD 405 that will be used in their inhaler device, ICO1. And ICO1 is the device that will then be used to administer the drug locally to the airways and the lungs. And that is a device that is simple to use, relatively cheap, and also with a high drug load capacity and a low loss of drug. In addition, it fulfills all the regulatory and quality standards and requirements that are needed for the clinical development. And in parallel, we are performing a market analysis in order for us to better understand the potential of RCD 405 in the drug market and to get an overview of the market in itself. And already now we know that there is a large drug market that is expected to increase even more, with about 5 to 6% per year over the coming decade. We also know that there is a high rate of undiagnosed patients, actually amounting to even 9% in some countries. And here we see the potential of diagnosing patients earlier, but also then initiate treatment earlier. So all in all, we have a very clear plan, and now with this additional money received from Vinnova SweLife, we see that the potential to progress the project at a faster pace is possible. And what the current aim is, of course, then to, as soon as possible, finalize the preclinical development phase where we are now, and also have the optimization of the formulation finished so that we can progress into the GMP manufacturing phase and prepare for the clinical development phase. And in parallel, of course, also then continue the business development and grow our company. So in summary, as you have seen from this presentation, COPD is a disease with a high unmet medical need, where there is a need for new and effective and innovative treatments. Here we see the potential for RCD 405, our drug candidate, which has a new and innovative and dual mode of action outside of approved pharmacological pathways. And it's also a compound with a long patent protection until 2039, currently in the major markets. And in addition, the market potential and commercial potential is large, and we see even the potential to grow this market even further in the coming years. So with this, I want to conclude my presentation and say thank you.
Thank you so much for your presentation, Mia. I'd like to start by asking you about the work done with one of your scientific advisors, Professor Eggestin.
Yes.
Together with him, you're trying to better understand the mechanism of action of RCD 405.
Yeah.
How would you say that project is going, and what's the ultimate goal there?
I mean, it's going fine, and now having received also this grant, which we will share between us, of course, we can do it at a little bit faster pace than probably anticipated from the start when we didn't have those money. But they are doing, and we, of course, depending on the results, we revise what to do next and a little bit trying to understand the pharmacology. And it's progressing, and I think we need to have all the results before, we can present anything more in detail. You have to get the overall picture.
Sure. And once you get that overall picture, what will that lead to? Potentially.
Potentially. That together with also some other results, of course, will then make us better understand at what concentrations we need to obtain to get the therapeutic effects. Of course, that will be not the clinical doses, but we can use that with these several studies to translate it and make a good prediction for the clinical doses. Yes.
And I was also wanted to ask you how the toxicology study is going, because I know there have been some delays in that study. If you could comment on that.
Yeah, we are having close contact with our CRO, and they will have promised us to send us and let us know of the results as soon as they have anything. Of course, they need to do the analysis and do an evaluation once the study is finalized, which it isn't yet, and to get that as soon as possible. But all the analysis and sample collection takes time, so it will be after summer. But we are in close contact with our collaborators and CROs performing this.
Well, I'd like to take a step back and look at the overall burden of COPD and respiratory diseases in general. You mentioned the fact that they're the third leading cause of death globally. Would you say that there's enough awareness around this?
Yeah, in some of the people working with it, of course, knows about it. But I think in general population, it's maybe not such a recognized condition or disease as other more like oncology and so on. So it's probably, I think there needs to be made more awareness of the disease. And as I also alluded to in the presentation, that a lot of people are actually undiagnosed, and they would benefit from being treated, especially with drugs that have an effect on the underlying disease that could stop the progression. They would benefit from being treated earlier and also diagnosed earlier.
Okay. Does this perceived, let's say, lack of awareness, does it affect the potential funding that would go into treatments for such a disease?
I don't know. The funding itself, for the development and for the studies that we are going, it's maybe a little bit more difficult and more competition for us than to compete with more oncology studies. But in the end, for the pricing and so on, there are rules and which are different in different countries. How that is, so I think that doesn't take into account the awareness as such of the disease.
Yeah, sure. Well, finally, I wanted to ask you, what are the main goals for Arcede Pharma in the rest of 2023?
Yeah, that we can progress the studies that we are performing and have a better idea of the pharmacological pathways, and then thereby also knowing the doses and the clinical doses, and get into the preparation and for the clinical phase.
Great. Well, thank you so much for answering the questions and for your presentation, and we hope to see you again at another event.
Yeah, thank you very much.